Perfecto. Entonces corroboro que estamos grabando en
Yeah,
español.
so I just wanted to confirm that we're recording in English,
Muy
Spanish,
buenos días.
and French.
Vamos a dar
Good
inicio a
morning,
este
everyone.
segundo día de taller sobre
We
buenas
will
prácticas
start
de
our second day
laboratorio,
workshop
conocidas como
regarding
GLPs, organizado por la Fundación de
GLPs,
Usos
or
Menores
Good
con
Laboratory
el apoyo
Practices,
del Instituto Interamericano
initiated
de
by the
Cooperación para la
Minor Use
Tecnología,
Foundation with
IICA.
the
En este taller tenemos participantes de América Latina y el
this
Caribe
workshop.
y
We
también
have
de África.
participants
Por
from
esta
Latin America
razón,
and the
estamos
Caribbean and also
realizando
from Africa.
el evento en esta franja
Therefore, we're
horaria.
having this
Tal y
event
como le
at
mencioné el
this
día de ayer,
time.
este evento se transmite en 3 idiomas.
As I mentioned
Inglés,
yesterday, this
español
event is
y
being
francés.
broadcasted
Por
in
lo
three
tanto,
languages,
hay
English,
dos
Spanish,
botones en la
and
plataforma
French.
Zoom que usted
Therefore,
puede
there are two
manejar
buttons
correctamente para que
on
vean
your
y
Zoom
escuchen
platform that I hope
en
you'll
su
be able
idioma de
to use
preferencia. Ayer,
in
los que
order
nos
to
acompañaron el día
listen
de
to the
ayer
conversation in your
recordarán que yo les
language
hablé
of
mucho
preference.
énfasis
Those of
sobre
you who
esto.
joined us
Es
yesterday
importante para que ustedes puedan
probably or may
ver
remember
y escuchar en su
that I
idioma de
uh,
preferencia.
insisted on this. It is
Aquí
very important for
podrán
you
ver un
to able to
botón
listen
llamado
in the language
idioma
of
de
your preference.
interpretación. Al darle clic a ese
On the
botón,
bottom
podrán escoger el
menu
idioma
bar,
que desean
you'll
interpretar.
see an
Esto
option, a
ya queda
feature that
publicado
says
en el video
interpretation.
especial
There you'll
y
be
espero que
able
ya en este
to
momento todos estén
choose the
escuchando
language of your
en su
liking
idioma de
that was
preferencia.
explained in the video, and I hope that
Para los
you're all listening
presentadores,
in the
les
language of
ruego
your
ser
preference.
consistentes en el
For all of
canal
those presenting,
Stream
I
en el idioma que hablan. Por
ask for you
ejemplo,
to please
si
be
presentan
consistent
en
in the
español,
channel
deben estar
in which you
conectados en el canal en español.
speak,
Esto es para evitar
in
ecos
which you
o
present. For
distorsiones
instance,
molestas en
if
los
you
participantes.
are presenting in
Las
Spanish, please
presentaciones
be in the
en
Spanish
PowerPoint
channel.
también se proyectarán en los tres
This will
idiomas
avoid echo.
y
The
nosotros
presentations, the
y
PowerPoint
sus
presentations, will
diapositivas.
also
Por
be
lo tanto,
shown
les
in all
solicito a
three
todos los
languages, and
presentadores
we will
indicar
manage the
la
slides.
palabra
Therefore, I
siguiente
would like to
cada vez que
request
desean
all
pasar a la próxima
speakers
lámina.
to say next every
El otro
time that you
botón está en la
wish
parte
to go to
superior
the
de la
next
pantalla,
slide.
llamado
The
Opciones de
other
visión.
button
Al darle
is
clic a
on
ese
the top
botón,
of your screen.
podrán
You will
ver las
see
tres
a
opciones
feature
de
that
presentaciones
says View
PowerPoint.
Options.
Podrán ver la
There you'll
presentación
be able
en
to see
español, inglés
all
y
the
francés.
options for the PowerPoint
Algo también que
presentation
deben
in
tener
English, in
presente es
Spanish,
que
and
cada vez
in
que
French.
hagamos un cambio
Something
de
to take into
presentación,
account is that
se puede
every time
desconfigurar
that
el idioma
we
elegido.
change a
Por tal
presentation,
motivo, si eso
you
sucede,
may need to reset
simplemente
the
vuelvan a
language of
manejar
your
esa rueda
preference.
y
If that
elijan el
happens,
idioma con
please
el que
go
quieren
back
ver
to
la
view
presentación.
options and choose the
El
language.
día de hoy contaremos con 6 presentaciones para
Today, we
cubrir
will
casi la
have
totalidad del programa.
6
Tendremos elementos
presentations
de
to
prueba
cover
y
the
referencia,
entirety of our program.
procedimientos
Elements
operativos
of
estándar,
testing and reference,
implementación del estudio,
operative standard operational
libro de datos
procedures,
de
study
campo, informe de
implementation,
estudio, almacenamiento y
field
conservación de
notebook,
archivos.
study
No habrá
report,
sesión,
and storage and conservation of files.
no habrá sesión de preguntas
We
después de cada
will not
presentación,
have a
solo
Q&A
hicimos
session
énfasis
after each
en
presentation.
este
That's
punto
something
el
that
día de ayer.
we mentioned
Si
yesterday
desean al final del evento,
at
si
the end of the
contamos con el
event.
tiempo
And
suficiente,
if we have
puede
the necessary
abarcar
time,
incluso las preguntas o
we will
comentarios que no
cover
tuvimos tiempo de ver
the
en su totalidad
questions or
el
comments
día de ayer.
that we
Durante
couldn't
las
address
presentaciones, si
yesterday
tienen
even.
dudas o
During
comentarios,
presentations,
vayan
if
de
you
una
have
vez
any
poniéndolas en el chat.
questions, any
Así
comments,
las
you
personas
can
que van
write
a
those on the
gestionar
chat.
las
Therefore,
preguntas
so that the
pueden ir
people who will be in charge of
gestionando las
the
respuestas
questions
en tiempo y en forma. Les recuerdo
can
que este evento será
give
grabado
you the
y que ustedes
answers.
tendrán la posibilidad de tener este contenido
I would like
disponible
to remind you that
en el
this
sitio
event
web
is
de la
being
fundación
recorded and
para
that
cuando
all
quieran
this
volver a
content will
repasar
be
la
available at the
capacitación
website
o
of our
enviarla a
foundation
algún
whenever
compañero.
you'd like to go
Durante
over
este
these
taller
materials or
estaremos
recommend them to
en
a
línea
colleague.
con un formulario electrónico de
During the
evaluación
workshop,
del
we
proyecto.
will be sharing
Les
a link
agradecemos a
with an
todos si al final del evento
e-form
se toman 4 minutos para completarlo. Esto realmente es
for
importante para
assessing our
nosotros
project. We
y agradecemos su
really
colaboración.
encourage you to take 4 minutes to
Esta información
complete
es muy
this form.
relevante
This
y de
is a
gran
very
importancia
relevant
para poder
information
irlos conociendo más y mejor acerca de
in
sus
order to
necesidades sobre
get to
estos
know
temas
you better
y
and what
para
your
poder
needs are
colaborar
in that
adicionalmente
related to
con
these topics, and
nuestros
to be able
aportes.
to improve on future events.
Vamos a iniciar ya con las presentaciones, con el
So
bloque de presentaciones
now
del
we
día
will
de
be
hoy.
starting with the
Y es un
presentations
placer
for today. And
presentarles
once
a
again, it is a
Javier
pleasure
Aguilar del
to
SENASA Perú.
introduce
Él
Javier Aguilar from
presentará elementos de
Senasa,
prueba
Peru. He will
y
be
referencias.
presenting
Sin más por el momento,
testing and
desde
reference
ya, Javier, muchísimas
elements.
gracias por estar con
Without
nosotros
further
y
ado,
dar esta
Javier,
presentación.
thank you very
Y
much for
le
being
cedo
with
la
us.
palabra. Adelante, por favor.
And
Gracias,
please
gracias,
go
Jorge.
ahead.
A ver, ¿me
Thank you.
escuchan
Thank you
ya
very much. Can you hear
al
me?
principio?
Perfectamente.
Ok, Eric, muchas gracias.
Yes, loud and clear.
Gracias al IICA, a la Fundación de Usos
Thank you
Menores,
to IICA, to the Minor Use
y a todos
Foundation,
los que vienen
and to
organizando
all those
este evento, evento
who
muy,
are
muy
organizing this event,
importante
a very
para todos.
important event for all of us.
Este es un evento que creo nos va a servir mucho
This
a
is
todos los
an event that
países
I think will help all of
por la
our countries
importancia que recae en todo el trabajo que se realiza
due to the
para la
significance
determinación
of all the
de
work
un
related
límite máximo de residuos
to establishing
radiactivos de uso
a
menor
maximum limit
que es de gran interés
of
para
waste for a minor
para
use
nuestros países,
system, something
para
that's of
nuestras
great interest
regiones, y eso nos conlleva
for
a
all of our
desarrollar
countries, of our
conjuntamente.
regions,
Todo este
and
trabajo
it
se
has
funda en el
led us
principio
to develop all of
de las
this
buenas
work
prácticas de laboratorio de la OCDE, con lo
abiding
cual
by the
permite justamente
GLP
la
practices of
determinación de este LMR
the OECD, which
que se
allows
desarrolle
for the implementation of this
de manera
EMLR
siguiendo todos los pasos importantes como los que se
to
ha ido explicando el día de ayer
follow
y
all the
lo que
important steps that
lo que se explicaron el día de ayer y lo que
that were
explicaremos el día de
explained
hoy.
during
En
yesterday's
cierta
session that
ocasión,
will be
¿eso lo
explained
pueden
today.
ver también en español o la tengo que
Just one
simplificar?
question. Can you see my presentation in Spanish as well, or do I
En
have
este momento,
to
Javier,
share that?
estamos proyectando la presentación en
Right now,
inglés, español y
we're
francés.
showing your presentation in all three
Ok,
languages.
bueno, siguiente, por favor.
Next slide.
Bueno, aquí tenía yo unas definiciones básicamente,
So
bueno, creo que ayer
I have some
ya se
definitions
mencionó lo
here.
que era buenas prácticas de
I think this
laboratorio,
was covered,
está
what good
relacionado
laboratory
básicamente
practices
con un
are.
sistema
This is
de
basically
calidad
related to
relacionado con los procesos
system, a
organizativos y
quality
las
system
condiciones
that is
bajo las
related
cuales
to the organizational
los
processes
estudios
and the conditions under
que
which
venimos desarrollando
these
y el
studies
medio
that
ambiente
we've been working
son
on
planificados,
are
realizados,
planned,
controlados,
carried
archivados
out, controlled,
y reportados. Ya Edwin también creo que ayer explicó
archived, and
gran parte
reported.
de
I
los
think that
archivos
Héctor
que se
explained
tienen que
yesterday
desarrollar
part of
siguiendo
this
todos los protocolos
archives,
de las
these
buenas
files,
prácticas
and
de laboratorio. Tenemos
everything that
que
needs to
en
be
estos
done
ensayos que realizamos
abiding by the
tienen que
principles of
registrarse. Y es
GLPs.
allí que
We need to
que
record
se define
everything.
o se refiere a un
And that's
artículo o una
how this
sustancia
testing,
que es objeto del
the
estudio.
test
En
item
esta
refers
sustancia o este
to
elemento
a
de prueba que es
substance
básico,
that is
no
subject
podríamos
of the study.
desarrollar un estudio.
Without
Por
this
eso que hay que
substance of
tener en
this
cuenta
item, we
la
wouldn't be able
definición
to
y
carry out a
también
study.
todas
That's
las
why we need to take
características
into account this
básicas
definition
que
and
se
also
requieren,
all of the necessary
sobre todo
characteristics
controlar para que esta sustancia de
required
prueba
uh,
cumpla con estos principios
or that
y
need to be
se
controlled
ponga
for
en
this
éxito
test
lo que
substance
queremos.
to meet all these principles and for
Lo
our
que
test
respecta a
to
elementos de
be as
referencia o
successful as we
control,
hope in that
los
related to
artículos o
reference
sustancias
element or
que sirvan
control.
para
This means any
evaluarse
article or
en
substance that
comparación
serves to
con los
give a
elementos de
basis for
prueba. Entonces es
comparison
muy
with the test
importante
item.
tener en cuenta los elementos de prueba y
Therefore,
las
it is very
referencias.
important to
Por
take into
favor.
account both the test and the reference items. Next.
Y es importante, como les decía, que se tiene que
As
tener
I
en
mentioned,
cuenta
it is very
la
important
recepción, la
to take into account
manipulación,
the
el muestreado,
reception,
el
handling,
almacenamiento
sampling,
tanto
and
de las
storage,
sustancias, de los elementos de prueba,
both
como
of
de la
the evidence
trayectoria.
and the
Es por
reference
eso
items.
entre la experiencia
That is why
que estamos
in
ya desarrollando nosotros
the
como Perú
experience that we've been working on here in
desde
Peru
el año 2019
since the year
con este
2019,
trabajo y iniciamos con un trabajo
which is when we
muy
started doing all
importantísimo
of this.
para el desarrollo de
We
un
started
límite máximo de
working
residuos
in
en el cultivo de
developing
papaya.
a
Todo
maximum
esto
limit
en
of
conjunto con
waste
Costa
in
Rica.
papaya crops.
Esto se
This
requiere
was
muchísimo
worked on
experiencia también
alongside
involucrada
Costa
y todo el
Rica.
trabajo que Costa Rica viene
Uh,
desarrollando fue también muy
all
importante
the
para
work that
nosotros.
Costa Rica has done was
Es
very relevant
interesante
for us. And
tener
it's
en
also
cuenta que debemos de llevar
important to take into
registros
account that
que se
we need to
mencionen las
keep
características de
records
los elementos de prueba
in which we
de
mention the different
referencia, la
characteristics
fecha
of
de
the
resección, la
evidence
fecha
and
de producción
reference, the
y las
testing and reference
características
items,
requeridas y
the
utilizadas
reception
para los
date,
estudios.
the expiry date, and the
Sería
amounts
preciso también
received, the
definir
quantities
los
received for the
métodos
studies.
de
It would be
manipulación, de
important also
muestreo
to
y de
define the
almacenamiento
methods
que
of
permitan
handling,
no
sampling,
solamente
and storage
mantener la
that allow
homogeneidad
to
y la estabilidad
maintain
en la
the
medida de lo posible para
homogeneity
evitar
and
la
stability as
contaminación
far as
o la
possible,
mezcla y también
all the while avoiding
recipientes donde
contamination
se
or
almacena
mixing, and
justamente
also storage
estos equipos de
containers
prueba y referencia
where this
deberán llevar
testing and reference
características
items,
de
they
identificación de
must
fecha,
bear the
de fechas
identification and date
específicas y las
information, the
instrucciones particulares
expiration date,
de
and the
almacenamiento que correspondan. Ahí
specific
pueden ver
storage
en la
instructions.
presentación en el lado derecho,
As you
tenemos
can
también
see
ahí la
on the
información que
right
se
side
registra
of this
de
slide, we
justamente
have the
la
information
recepción,
that is
almacenamiento
recorded
y el
of
desecho también que corresponden a las
reception,
instancias de prueba.
hand
En el año
storage,
2019
and also
trabajamos justamente
waste
con una
in
sustancia
terms of the
active
substance, the
luminescensation
testing substance.
que
In
prácticamente
2019, we used
muestra
an
toda la
active
información. Aquí
substance
está el
called
número del lote,
luminescensation. Here
la
you can see
fecha
all
que
of the
recibió
information, the number of the lot,
el
the
elemento de
date when
prueba.
the
Bueno,
testing item was received.
es un formato de recepción
This
y el
is a
transportador por
reception
estancia de
format.
prueba, la persona que recibió
It
esta
says
sustancia de
who
prueba,
transported
la guía, la
this
factura o
uh,
un
testing
documento
substance,
del
number of the guide,
fabricante,
the
cuál
invoice,
fue la cantidad aproximada que hemos
what
recibido,
was the amount,
los recipientes, en qué presentaciones viene este recipiente y las condiciones también de los
also
recipientes.
the conditions
Asimismo, también las
of this
temperaturas
container,
mínimas de
and
almacenamiento
also
de esta
the
sustancia de
minimum
prueba y las
temperatures
temperaturas
for
máximas
storage of this
que
testing
corresponden a
substance, and also the
acuerdo con el
maximum
protocolo que
temperatures
más
that
adelante también se les va a
correspond
explicar.
to
Siguiente,
the
por
field
favor.
notebook that we will be explaining afterwards.
Es importante que estos elementos de
It is
prueba
very important for all of
o de
these
referencia
testing
estén
items
acompañados con un
or reference
certificado
items
de
to
análisis
come
para
together
realizar la
with an
aproximación
analysis, a certificate of
que
analysis
tiene a cargo
that is
este
provided
elemento de prueba. Y es importante
by the
conocer también
sponsor
que
in
además
charge of
tienen un
this
certificado
testing
de
item.
análisis,
And it's
como lo
also important to know
mencionábamos
this
en la
the information
presentación anterior,
contained in this
en el formato
certificate
de
of analysis.
recepción.
As I
Toda la
mentioned before,
información corresponda o venga establecida en este certificado de análisis, que es
all the information
verdaderamente
contained
que nos va
in the certificate of
a conducir a que el
analysis
ensayo se
will be
desarrolle siguiendo estos
that
principios. Como
what
pueden
will
ver
allow
aquí
us to
en el formato
abide by the
de este
principles
certificado de
during
análisis,
this
viene el número de
test. As you
lote,
can see here in the
toda
form,
la
we have the
información
number of the lot, we have
de
all
referencia
of the
del
information
propio producto,
of the
¿verdad?
reference
Tal,
information of the product
por
or
ejemplo,
substance.
en la pantalla al lado
For
derecho
instance,
pueden
here
ver la información a quién
to your
viene
right,
destinado esta sustancia
you
de
can see
prueba y también es importante también
who this
conocer cada uno de los
as
países donde está
test
esta
item is
masa
being
que
sent
corresponde
to. And
justamente a
it's
que este
important to know
producto
all the
pueda llegar
mechanisms
a
in different
término
countries that allow
siguiendo
for
todos
these products to
los
reach
controles
their
y
destination
no tener problemas al momento
with all the
de
controls so
desaduanar.
that
Entonces
they
nosotros
don't have
en la primera
problems
experiencia
while they're
este
in
producto
customs.
llegó
And during the
sin ningún
first
problema.
experience,
Este
this
año también
product
recibimos otro
had
producto
no problems getting to its destination.
desarrollado en
This
otro
year, we
continente
received
y
a
nosotros
different item
seguimos todos
for a
estos
test
mecanismos
in a different
de
kind of
control que
crop,
tienen
and we
establecidos
followed
aduanas
all
para
of the
recibir
control
justamente
mechanisms that
estos
customs have
productos y
in order
tenerlos
to receive these types
listos
of
para
products and
desarrollar
have them
nuestros
ready
ensayos. Siguiente, por favor.
for our testing. Next.
Bueno, acá para nosotros como Perú tenemos un decreto supremo que es el
For
decreto
Peru,
supremo número
we
001
have a
2015
supreme
Minagri
decree,
y
which
está
is the
referido al registro
001
de todo lo que
2015
está en sí
Minagri, and this
relacionado al
refers to
plaguicidas,
the
control
registry
agrícola
control
y
and
también
surveillance
tanto químicos
for
como biológicos.
agricultural
Y
plaguicides, both
este
chemical and biological. And this
en una
supreme decree,
de
in
sus
one of its
disposiciones complementarias, establece también
complementary
que pueden ingresar
provisions,
productos para
establishes
uso oficial y es
that
por eso que nosotros
products
tenemos
for official
la
use can enter. And
caracterización
that's
del
why
producto
in
y le damos
Peru,
un permiso para la importación, el
we
cual
issued an we issue
se le da
an
toda
import
la
permit
agencia
for
de
these
aduanas que
products to
viene
enter our country.
realizando este
We
trámite de
share it
importación
with the
y como
customs
esto
agency
permite justamente que el
for
producto
all of the
llegue
import
hacia
procedure,
nosotros
and this
sin
allows for
ningún
the
problema.
product to reach us
Lamentablemente este año
without problems.
no con el elemento de prueba, sino con el producto de
Unfortunately,
prueba,
this year,
hemos
not with
tenido un
the
problema, pero
testing,
eso
but
fue
with
un
the
problema
reference items,
en
we've
otros
had a
países
problem,
que
but
no
these
nos ha
are more
permitido que el
related
producto
to
llegue
control
todavía a nuestro
issues in
país.
other countries
Hemos
that have
solicitado todo el
allowed
permiso
this
nuestro para que el
product to
producto
reach
ya
our country. We
desaduanado
have met
en
all the
Perú. Sin
paperwork
embargo, ocurrió otro problema
for
que
it to
está todavía en
arrive here
investigación.
in
Estamos
Peru.
buscando solucionarlo
However,
para
there
llevar
was a
la
different issue
referencia
that is being investigated, and we
hacia
hope that
nuestro
this
país.
reference item
Es para continuar
will
ya los
reach
trabajos
our
en
country
laboratorio.
in order
Sin
to
embargo,
continue our lab work.
con toda esta
Nevertheless,
normativa que tenemos
with all of these
en
regulations that
Perú,
we have
nos
in
permite
Peru,
que el
thi-
producto ingrese o desaduane
this allows for the
sin
product to
mayor problema
enter
y que el
and pass
producto
through customs
se pueda solucionar y
without facing major issues.
comercializar
We hope
la
to be
sustancia
able to
de
solve
referencia
the situation and to have this
Lamentablemente,
reference item as soon as
esta es
possible.
una
As you can
foto
see here,
justamente
this is
del,
a photograph
del envío. Entonces ahí viene toda la información. Por eso es
of the
importante
shipment. It has
al
all of the
transportador enviarle
information.
la información correcta
It's important to
porque
send all the
puede
correct
evitar
information
justamente
to the
desaduanar sin
sponsor,
mayor problemas. Estos
since this will
productos que hemos
allow
enviado en
for it
estos
to pass through
ensayos
customs
que
without
venimos
facing
desarrollando
major issues. These
ha mantenido
items
justamente
that we
el
have
costo
received
y ha
for
mantenido
these two
la
tests that
información
we're working
correcta
on,
y
uh,
esto nos ha permitido
we have kept uh,
desaduanarlos acá en nuestro país. Siguiente, por
our
favor.
information in an orderly fashion, and that has allowed us for them to pass, or it has allowed these products to pass through customs without facing major issues.
Bueno, entonces acá también es importante conocer la caracterización.
So it's very important to know the characterization here. And what the good laboratory practices tell us is that any evidence or reference item must be properly identified, which means its code, its registration number or CAS number, the biological parameters. And as you can see there on your screen, on your right side, we have the lunar sensation, and it has the CAS number and all the information related to the product. So it is critical to take into account all of this and to also coordinate this with the sponsor so that all of this information is included in the documents that are sent along with the product. So that we can have all of that information and record it on our documents. For instance, our field notebook. For each one, for each study, it's important to know the exact nature of the testing and reference items, and specifically the number of the lot, the purity, its composition, concentrations, and other characteristics that allow to define each batch adequately. When a facilitator sends the testing items, there must be a defined mechanism alongside with the sponsor that allows to verify the identity of the item. It's very important to follow all of this information related to the characterization because this will allow us to develop our studies abiding by these principles.
Sí.
Next.
Acá, como pueden ver, está la información
As you can see
relacionada
here,
al producto
we have all of the
de nuestra
information
referencia. Vemos acá
related
la
to the
información
reference
que nos
item.
compartieron. Y también, bueno,
This
en
is
esta hoja
the
práctica
information that
que
was
es el libro de
shared with
campo,
us.
también
And here
registramos
in our
justamente el uso,
field notebook, we also record the
cuando hemos
use
iniciado con nuestras aplicaciones.
when we
Por eso es
started
importantísimo
applying
contar
the
con
item.
esta
And that's why it's
información.
so important to have
Entonces,
all of
esta
this
información nos permite
information
justamente
because it allows
identificar y
us
cumplir
to
con todos
identify
los
and
pasos
to
necesarios para
follow all
desarrollar
the
de la manera
necessary
más
steps
adecuada nuestros estudios
in
y
order
cumplir
to
finalmente
perform our
con lo que
studies
indicaba
in the
anteriormente
best way possible. And
y,
also
y
to comply
determinar
with
y
what
establecer los
was
niveles
mentioned
máximos
before,
reconocidos
to
siguiendo
determine and
estos
comply
principios.
and
La
establish
siguiente, por
the
favor.
maximum required limits.
Bueno, otros puntos interesantes también dentro
So
de
other
caracterización es
interesting
para
points
todos
in
los
that related to
estudios
characterization is
es
that for
preciso
all
conocer
studies,
debidamente
it is very
la
important to
estabilidad de los
know
elementos de
about
prueba
the
y de
stability
referencia
of the
en
test
las
and reference
condiciones de
items
almacenamiento
under
y de
storage
pruebas,
and
también
test
si
conditions,
se administra o aplica en
whether
un vector
these
como
are being
elemento de prueba, será
applied or
preciso también
administered
determinar la
to,
homogeneidad,
to a
la
vector, it would be
concentración
important
y la
to determine
estabilidad
the
del elemento de prueba
homogeneity, the
en ese
concentration, and the
vector.
stability
Para
of the test
elementos de
item in
prueba
this
utilizados
vector.
en estudios de campo,
For those
como
test
por ejemplo
elements
los
used in
depósitos,
field studies,
esta
such as
información se puede
mixtures,
obtener por medio de experiencias de distinta
we can
índole
obtain this information
efectuadas
through
también en
different
laboratorio.
experience
Y una muestra de cada
carried
lote
out in the
de elemento
laboratory,
de prueba
and a sample from
se
each
hará de
lot
colectar
of this test
para
item
fines de análisis para
should be
todos los
retained
estudios,
for
excepto
analysis
tratar para
purposes,
estudios a corto plazo. Entonces es muy
except for
importante
short-term studies.
conocer también estas características y
So
desarrollarlas
it's very
los
important to
ensayos
know
a
all of
corto
these
plazo.
characteristics
Nosotros durante
so
todos
that we
estos
can
dos ensayos
perform our tests
y este
in a
curso que
proper
estamos
way.
desarrollando,
During
he
this test
recibido
that we've been
muchísima
working
ayuda
on,
conocer justamente toda esta información
it,
para
it's been great for us
llevarla
to know
a
all of
lo
this
que
information so
actualmente
that we can
desarrollamos tanto
apply
en
it
campo y
to
de esta
everything
manera,
that
bueno,
we're
lo vamos a desarrollar también
doing in the
en
field.
laboratorio. Por eso es muy
And
importante
this year, especially
muy
for everything that
importante
we will be doing in the lab. So
tener en
it's important
cuenta
to have all of
los
this
recursos que
information
requieren
available to take
estar
into account
vinculados a estos elementos de
all
prueba
the
y de
records
referencia
that need to be
y de
kept
esta
in
manera
terms of this
cumplir
test
justamente
and
con los
reference
principios
items,
de las buenas prácticas de laboratorio.
therefore
Siguiente,
meeting
creo
all the
que ya estoy
principles
finalizando ya
of the
mi
good
presentación. Es una
laboratory
presentación
practices.
corta,
Well,
pero
my
tal vez
presentation is coming to an
permite
end. This was a very
justamente
short presentation, but one that
seguir
allows
cumplir
us
con estos principios, desarrollar los
to
estudios,
follow all of these
siguiendo con
principles
todas estas recomendaciones
when
establecidas
performing these studies
por
and
la OCDE y de esta
applying
manera
all of these
cumplir
recommendations that have been
con lo que
established by the
establece o con lo que
OECD
se establece para tener
and to
tener
comply
un límite máximo de
with
residuos
that
cumplido
established
con
for
todos
maximum
estos
residue
principios. Muy
limits
agradecido por esta invitación, por esta
while abiding by
presentación
all of these principles.
y
I would like to thank you all
darle las gracias a
for
todos
listening to this
por haberse
presentation.
unido
And also thank you
temprano
for
en Perú.
joining us.
Son
Uh,
7 de la mañana en Costa Rica, creo que es
it's
6 de la mañana.
7 a.m.
Y
in
bueno,
Peru. I think it's
agradecerle
6
y
a.m. in
aquí
Costa Rica.
continuarán
So
las
I would like
presentaciones
to
siguientes.
thank all
Muchísimas
of you
gracias.
once again. And we will continue with our presentations. Thank you very much.
Muchas gracias, Javier, por tu presentación. A
Thank you,
continuación,
Javier,
vamos a pasar a
for your
a la siguiente
presentation.
presentación del día de
We
hoy,
will move
a
on
cargo de Carla Arrieta,
to
del
our
Servicio
next presentation
Fitosanitario
with
de
Carla
Costa Rica,
Arrieta
con el tema procedimientos operativos
from Costa
estándar.
Rica,
Aprovecho para
Phytosanitary
agradecer a todos los que
Service
han
in
estado
Costa Rica
escribiendo en el
with
chat
the standard
el
operational
nombre,
procedure.
su país y su
So
organización.
I would like to
Los que
thank
no
all
lo
of
han hecho,
those who
les
have been
agradecemos muchísimo.
sharing in the
Gracias,
chat
Carla.
your
Por favor,
name,
escriban estos
the
datos en el chat
organization that you come
para
from.
referencia
For
nuestra.
those of you who haven't done it yet,
Carla,
please do
bienvenida
write this in
nuevamente
in the chat so for our reference. Carla, welcome once again. Please go ahead. Well, good morning, everyone. Thank you for sharing with us today. I hope that this will be a very productive session. This is a very short summary of operational standard procedures that are used for in good practices. Okay, let's wait for the presentation to be shown on the screen.
Bueno, yo trabajo para el servicio, para el laboratorio de análisis de residuos de plaguicidas del Servicio Fitosanitario del Estado. Y nosotros tenemos ya bastante rato.
I work for the Residue Analysis Lab of the Phytosanitary Service, Costa Rica. We support studies for a maximum limit of residues in minor crops. We started our service with the implementation of the good agricultural practices, and right now we're working on the toxic coffee study, and we're at the laboratory stage. We have had we have been for a while using this good laboratory practices and appropriating these good laboratory practices, because as if it was mentioned before by my colleagues, this is a quality system that takes time, as any other quality systems, but through time you see the results of working with a system like this one. In the case of the standard operational procedures, this is what we use to record or describe every test or activity that will be performed. This means that we need to have a procedure for each activity every time that we have a routine activity in the context of our study. Could you please go to the slide after the next one? So we define every routine activity as every activity that we have performed previously and that we need to perform again in the exact same way. And most importantly, for all people to know exactly how it needs to be performed. Carla, we will stop very briefly. It seems like the presentation in our English channel is frozen, so we will stop just to solve this technical issue.
Yo estoy mirando en el canal de inglés la presentación.
Looking at the presentation in the English channel, and I see the presentation of Veronica Picado. Maybe if other colleagues see the same thing. Yes, correct. In the English channel, we're seeing presentation of the files, the archives. So we will wait for the presentation to change. Yes, it's correct on French. Well, I am seeing that we no longer have the presentation available in English. Now we are seeing the correct slide. Very well. We have English, Spanish, and French, and we're in the correct slide. Carla, you can continue. Verónica, Adriana, I think we lost Carla. Maria, can you confirm if Carla is available? I think she's getting online. Yes, she's coming on back again. You're in mute. Yes, forgive me. Sometimes I have issues with the internet. You're here again with us. Great. As I mentioned before, well, basically, these SOPs facilitate to give order to every routine activity taking place within a study. Next. So why is it that under the OECD principles, they indicate that every trial installation must have its own SOPs or operating procedures? Well, these studies try to execute the activity always in the same way, that there's no room for interpretation. And the matter of standardizing everything in a way guarantees that the data being obtained is valid and reliable at the end of the day. Also with this, we avoid improvisation, that people do things differently and sometimes we'll be doing it one day one way and changing it the next. So the study doesn't follow a logical axis of these activities, and it might be that at the end of the day, the results being obtained are not reliable. And also with the procedures, we indicate the people their responsibility. So we assign every person the activities that they need to perform. Next. According to the principles, the SOPs need to have some minimum requirements where all trial installation or facility must have its own SOP or those given by the sponsor or the study director. With respect to this, the IR4 has some templates for registration or certain procedures that might be used for the implementation of best laboratory practices. But in my experience, I would say that as much as possible, try to elaborate your own SOP because it's not the same thing to use documentation elaborated by another person than when you do that yourself. In this case, it was in our experience when we began with the implementation, we began using the ones from IR4, but when we put them in practice, it turned out that we could not really understand what's the information that we needed to put there, or we considered that maybe data were missing that need to be written down, or even that there were things that did not apply to us. So that's when we began to develop our own SOPs, and we got to a point where we have them pretty much advanced, working independently or of the study that's taking place, because we'll see further on the SOPs cannot be too general or not, or too specific. Another important thing with the SOPs is that they always must be available for all the staff in the study. They must be in a place that's accessible for all staff, and important that everyone has a copy in their workplace. Another important thing is that the SOPs must be elaborated by the study staff, by the same people developing the activity, and these should be approved by the facility director, both to ensure the quality and the integrity of the data. This approval must be made in writing. It must have a signature and a date. Next. any change or deviation that there is from an SOP that's been established must be communicated in writing and also must have the approval of the study director. Many of these requirements have been mentioned in some of the previous presentations. Nonetheless, it's good to have this present in case of any deviation or change. This must always be communicated, and all the documentation generated under the good practice must have unequivocal communication being managed, in our case at least, using a document, what we call document control. And here we have a list of all the documentation that's being generated for the studies that are performed under good laboratory practices. Also, the principles of OECD say that we can make use of books, handbooks, manuals, or analytical methods as a supplement in the elaboration of the procedures in each one of the facilities. Next. And this is what I mentioned before. Every SOP must have enough detail for anyone to be able to follow them and generate good data, that there is no doubt of how these activities should be performed. And in this way, to guarantee that the study director and the facility director can give traceability to how these activities took place, whether it's in the lab or in the field, because generally neither nor the director of the facility or the general director are looking at what everyone's doing. So with these registration process, we can ensure that they can recreate the scene of how these activities took place. And the fact that they cannot be too specific. So if we have a POE that's too specific, this might turn out with deviations. So we need to communicate to the study director because we did not follow the SOP as it was written. And this was quite complicated to us. At the beginning, we created certain SOPs with certain registrations, but at the time, we created them for a polypoxetan study in bananas. So the SOPs worked very well, but when we began to perform the study in papaya, we noticed that there were many things that these SOPs had that did not adapt to the study in papaya, or the fact that for banana, for example, we separated the pulp and the skin, and papaya was a sample that was processed as a whole. So we started to think how we could allow these procedures to be general enough, but with enough detail, and at the same time, they were specific enough for each study. So we did not have to renew all the documentation every time we had a new study because of the things that did not apply. So in this case, we thought to do a general procedure, for example, in the case of the sample homogenization, where we put this in general terms, that the sample need to be homogenized, maintained in a cold storage with a certain temperature, these type of things. So this procedure got a registry added or made reference to a registry created specifically for this. So in this registry, we put specifically how the process took place, whether it be papaya, banana, or the ones we have worked with, such as coffee. So the registration, as it is completed at the time with the information that we have available according to the study taking place, then it gives us that flexibility of not having to modify those SOPs every time there's a new study. And with the registry, we can be specific enough with respect to the activity that's taking place at the time for that study specifically. The other important part with SOPs is that we need to train the staff. with respect to the documentation that you have within the system, and also for new staff and existing staff that is already part of the study, you need to train them as well with a refresher course. Next. The question then becomes is how do we begin elaborating an SOP? Well, for me, this is the central axis of good laboratory practices, because if we don't have a procedure that indicates how these practices should take place, then we are basically doing this blindly. The protocol has certain aspects of how these should take place. For example, the rate of application on a field, or that the lab needs to validate 2 or 3 levels with certain amount of replicas, but it doesn't say specifically how the chemicals should be prepared, of how we should grind the sample, or in the case of the field, that you need to control the flow, the application, and the mouthpiece of the pump. These things are just up in the air in a protocol. So that's the importance of an SOPs. So when we elaborate these SOPs, these need to be easy to understand for everybody. And as I said before, it cannot be too general or too specific. We make use of manuals and guidelines for the elaboration of these documents, and with a control list to have some sort of traceability. And something that's also important is that SOPs and registries are dynamic documents. So we need to keep updating them constantly and revising a certain, well, every certain amount of time. It's not a static document that once they're elaborated, they stay like that for their life. But as I mentioned in our case, we are in around version 3 or 4 of the SOPs, because we began this process. It has been slightly perfecting itself through time. To begin with the elaboration of the SOPs, what's most important initially is to elaborate an SOP where you have a format for the document. In this SOP, we are indicating what sections the document should have, what's the format where it should be elaborated, what type of coding it will be given for each type of the procedures and items, and also define the language, the type of font, the size of the font for all the documentation. Another important thing is a date format, because we know that there is a North American format. We know that there is a format of what we use in Latin America that is different. So it's important to design a way that the date is stated in these documents, if it's the month or the day first, to avoid confusion. Month, day, year, and not vice versa. It's also very important the way how the coding of the information will take place. Next. For example, in our case, we use this coding format, and we have changed this maybe 3 or 4 times, because first it made reference to IR4. So then afterwards, since we don't work with IR4, but minor use foundation, and also IR4 only gives us some directives, some general terms. There's no reason to link it to a coding system. So we have changed this 3 or 4 times, and this is what we're using currently, where we have PMRP followed by 2 Ys and 2 X's. Well, PMRP corresponds to the projects taking place establishing residues, maximum limits for these pesticides. YY corresponds to the type of document, whether it's an SOP, R if it's a registry, or EX if it's an external document. And XX corresponds to a consecutive number to each document. Next. And for example, in the structure of these SOPs, we define that they have 5 main sections, where first we indicate the purpose under which the procedure was created. Second is the reach. If, for example, the SOP is trying to reach homogenization, and then be very specific about how these apply, and then we'll have a description of the activities that need to take place, such as a logical sequence. And then we have the annex section where we have any complementary information. For example, in this case, we can write down the reference method that indicates the protocol, for example, in the case of the sample analysis. And very importantly, a section of control change where we indicate all the changes that the document has had throughout its life cycle. The part of change, we manage that as a separate registry. So basically in the SOP, we make reference to this other document, because otherwise we would have too many pages within the document just indicating the change, control change. And these need to be written down somewhere. So that's why we generated a new registry to write down these changes. Next. And with respect to the principles of the OECD, they require that they must have at least these SOPs. Many of these have been mentioned throughout the presentations before me. And for example, as Javier mentioned, any trial facility must have a registry and SOP for the management of the samples and the trial substance, how these are identified, how these are received, how these are labeled, how they are handled, whether it be in the lab or on the field, how the sampling will take place, and how it is stored, whether it's inside the lab or in the field. In the case of equipment, the materials and the reactants, they must have calibration, maintenance, cleaning of any of the equipment used. With respect to information systems, these must be validated. So you must have SOPs where we define all these directives, both in operation, maintenance, security, backup. In the case of information systems, also, you replace or change someone in the staff. And in the case of the equipment used for data analysis, generally, we have access passwords with only certain people can have access. So how do we change in case that we require to change the person responsible? With respect to materials, reactants, and solutions, we also must have SOP for their preparation and labeling process. In this case, within the lab, we have a system under ISO 17025. So with respect to these certifications, we share certain characteristics or principles under which we have use and maintenance, and that has to do with equipment calibration and maintenance. So we had already implemented this for ISO certification. So what we did was to adapt or to make reference. In the case of the equipment being used, we made use of the equipment that had been established in the ISO certification. With respect to the lab, just not to duplicate or do work that the laboratory had already established. For example, everything that has to do with use registration and calibration of the equipment. Next. we also must take into account how we maintain or we store the registration and how we compile and store the data. And also, this is one of the following presentations of how we will archive the data being generated. And also very important, we must define how we will code the studies within the trial facility. And as Veronica mentioned, the part of insurance, quality assurance must have its own procedure. So these are the primary points under OECD principles. It says that any trial facility must have SOPs, but we must not limit ourselves to these. Every facility or every work group develops their own SOPs according to their own criteria and needs, as long as you have at least the ones I just mentioned. And I think basically that sums up standard operating procedures, and I hope this is useful. Also, we have the best disposition, willingness to help you if you have any issues elaborating your own SOPs. Next. And this is the end for me. Thank you very much for your attention.
Muchas gracias, Carla.
Thank you, Carla, for your presentation. I remind you that your questions can be written down in the chat box so they can be analyzed and passed on forward. I thank you to the people that have put their name, their country, and their institution in the chat as well for our own reference. The third presentation today comes from Edwin Barbosa from Agrosovia, Colombia, with a principle of the implementation. Edwin, the floor is yours. Hello, Eric. Good morning. Can you hear me? Yes, we can hear you. Great.
Qué bueno poder verlos. Bueno, permítanme un segundito.
Please give me just 1 minute.
Correcto. Ok. Bueno, muchísimas gracias
Very well. Thank you very much for the time you're spending in this workshop yesterday and today. I will tell you about the execution of the study, the implementation of the study. This is information that we prepared together with Ken. Nonetheless, I will present it, but the information here was prepared between both of us. So with his permission, I will tell you a little bit about how the study takes place for establishing these maximum limits. We will see 3 specific topics. The first has to do with the study plan that we all know as protocol. The second is what is the content of that study plan? Within its structure, what are the primary aspects that must be present in that study plan to be able to develop those maximum residue limits? And the third aspect has to do with the in situ implementation on the study plan. So let's begin with the first one. Let's talk about the study plan then next. Well, when we establish these studies for establishing maximum residue limits in a specific product, the first thing that we must build is a plan, a study plan, or what we also call a protocol. And here I want to highlight the fact that this study plan is the main document of the whole study. Here we compile everything that must take place both on the field and in the lab, and what are the specifications for each one of the topics that are being developed both on the field and on the lab. The specific activities that derive from the laboratory activities and field activities will be a little bit more specific in the SOPs that Carla just mentioned. So basically, the study plan is what will describe the procedure. Since this is the main document of the study, it's the one that gives specifics about what should take place throughout the study. And any change present there is a change that must be reported to the study director beforehand. And we will see a little bit more in detail what this entails. These two situations that might take place. But what I want to highlight is that this study plan is basically a roadmap that we must follow in the study. And here we describe how many trials will take place, who will perform them, and if these are standard studies where we apply the pesticide with a pesticide and a determined dosage and taking samples at a determined date, or if there are other trials such as processing where we evaluate different parts of the crop. For example, if we are looking at the skin of the fruit, if we're going to evaluate residuality on the skin or the pulp or the fruit as a whole, and also a third type of trial, which is a trial that looks at application with the frequency and dosage mandated by the protocol. And here we can see the samples taken at different time frames in order to see the study and the behavior over time for the pesticide.
This study plan must clearly describe the objective of the study and the experimental design for performing the study, or how we will do it, how many untreated parcels we will have. Typically, it's one. How many treated parcels will we have, and what are the types of tests that we'll have? So in this study plan, we include everything that needs to be done. This study plan must be easy to understand, must be known by all the personnel involved in the study, and the people involved in the study need to be aware that everything that's included there is what needs to be complied with. When we're going to start a maximum residue limit study, the plan needs to be approved and signed by the director of the study and also by the sponsor. This needs to be done prior to the to starting the study. The starting date is the date when the protocol is signed. From that point onward, the study will apply. So for instance, if I will apply it today, the protocol needs to be signed by the director of the study with today's date. Why? That because this is the roadmap that we will follow, and this roadmap needs to be approved by them. This protocol has to be verified by the quality personnel. They don't approve what it's written. what they say is that this protocol meets the good laboratory practices. What is the objective of these study plans or protocols? First and foremost is for all the personnel involved in the study to know the objective of the test, why the test will be performed, for them to know also the magnitude of the test and how it will be performed and what are the different things that we need to take into account during this study. As I said, this is the roadmap that establishes how the study will be performed. It also determines when and where the study will take place. We will discuss a little bit about the dates. The protocol needs to include the starting and the ending dates of the study. It also indicates the person in charge of the study or the study director, who will be the person who's in charge of the activities, the field activities, but also the laboratory activities. And it's also the person who's in charge of making the decisions whenever there's a situation in which what's written cannot be complied with, or there's any deviation or something that it's outside the control of the personnel. It also mentions who will participate in the study. So in this plans or protocols, it says who's the study director, who's the sponsor, who's the leader of the quality assurance, or the person who will be in charge of that unit. And it also mentions two very important people for the study, who are the field investigator, who's in charge of, the field researcher, and the lab researcher. Who are the people, or who will be the person in charge of everything that's related to the analytics? This study plan needs to be robust enough for it to be a foundation for the building the study, meaning that any person who wants to read the study plan can easily understand what was done whenever the study was performed in a given country or a number of countries whenever there's collaboration involved. Edwin, I'll go to the next slide soon. I think that someone said in the chat that someone was having problems with the interpretations. I just wanted to confirm that the interpreters are in the channels where they need to be. And I would also like to tell the participants that on your lower menu bar, you can find a feature that says interpretation. It's a globe. It says interpretation. There you can choose the language in which you would like to listen or to follow the conversation. And if you go to the upper side of your Zoom platform, you can see view options. If you click there, you'll be able to find 3 options in order to see the presentations in English, French, or Spanish. Again, this is for the PowerPoint presentations. I hope that I clarified this. I'm sure that the interpreters have uh, confirmed that they're in the right channel. So Edwin, we can move on. The study plan describes with enough rigor the study that we will be performing. It allows for the reconstruction of the study, but it can also be audited, as I mentioned, because the study plan needs to contain the principles of the good laboratory practices. This needs to be an easy to use document, and it can be easily located, which means that we need to have this protocol in the field and in the lab, and the people involved The studies need to really know this studies. One of the responsibilities of the study director is to have, for this protocol to have, for the field researcher to have the study at hand, and for them to share it with their work team, with their analysts, in the case of the lab, or to the people helping in the field. Since this is the roadmap, it is understandable that this roadmap should have a controlled distribution. So there are official communications between the study director and the lab and field researchers, so that there's a single version, that's the one that's being worked on, and to make sure that this version is completely controlled by the study director. This protocol must contain between 20 and 25 pages, and it's identified with a number. This is, you can see the number here in the upper right side of the page. We will discuss this briefly. Okay, for those of you who are new to this type of tests and studies, it is very important to have clarity about what we'll be discussing. Sometimes it's a bit complicated, but once you learn it, it becomes very, very easy. This protocol can have amendments or changes to it, but it can also have deviation. What's an amendment? It's something that I will change in the protocol in terms of what was initially written, but I do this prior to the beginning of the activity. So let's say that I said that we would have 3 tests, but now we're going to have just 2. That's an amendment that I'll make. The initial protocol is the main roadmap. And whenever there's a change made to it, these changes need to follow certain specifications. So again, this amendment is something that I change before starting the activity. So to change anything, I need to justify what I would like to change this. And this needs to be justified, but also needs to have date and sign and signature of the study director, because generally it's the study director, the one who manages any amendments to the protocol. All of these amendments need to be conceived as an integral part of the protocol. So we have the initial protocol, and any amendments will become part of the protocol from that point forward. They need to be described and explained by the people responsible of the study, and they need to be signed with date by the study director. So there may be an amendment to the protocol. Let's say the field researcher saw that there was something that couldn't be complied with for any given reason. This person conveys to the study director that they won't be able to comply with what the protocol says, and that they would suggest to do a different thing. And it will be the study director, the one that makes the decision on whether this amendment is justified and how the new specification of this activity would look like. It needs to be clearly described. This one is very small, but it's an amendment. An amendment has a number. This will allow me to know that I have my protocol and the number of amendments that it has. It needs to have a title, the title of the project that it's being amended, the number of the project, the description of that amendment, how it was before and how it will be now, and what is the reason behind this amendment. This will allow me to ensure that all of these changes will be controlled and that they will be justified. This and that related to the amendments. Again, this is something that I changed before starting the activity, and this is something that does not follow what was written in the initial protocol. Now, what is a deviation? A deviation is something that occurred and that does not comply with the protocol. This is something that we hope will not happen, because it would mean that we wouldn't be complying with what was initially written, but it's something that can happen, whether due to weather conditions or because the specific SOP wasn't read and the activity was performed in a different way. So therefore there was a deviation. We need to report those deviations, and this will be very important for the studies, because this is something that should have been complied with. It wasn't complied with, and we need to report what the reason why. This is a format of the deviations. This is included in the field notebook.
Perdón, el investigador de campo
The field researcher can fill out the deviations that took place during the study. Again, hopefully there won't be any deviations, but if they happen, we need to document them. They must be dated. We need to include the date in which the study director was notified, hopefully as soon as possible, so that if any measures need to be taken in terms of those deviations, the study director can be able to do so. We also need to mention the method in which the deviation was notified. Sometimes it can be a call, but this call needs to be registered in writing or recorded in writing. There can be deviations from the protocol, or there can be deviations from the SOP. For instance, I needed to wash the equipment with a 0.4 ammonia solution, and I used a 10% ammonia solution. So that's a deviation from the SOP. Let's say the protocol says, do not apply the test substance below 32 degrees Celsius. And while we were in the field, let's say that the temperature was 34 degrees Celsius. So that would be a deviation from the protocol, because the protocol said, do not apply this substance above 32 degrees Celsius, and I would need to inform the situation so that the study director takes the corresponding measures. These situations need to be sufficiently explained, why they happen, how they happened, and they need to be validated by the study director, because it will be the study director, the one who will say this can have a low, medium, or high impact on the results. So it will be the study director, the one who will assess, who will sign, and who will date that assessment, and will also notify the sponsor so that they can sign and date. Obviously, these deviations and the amendments that I mentioned before need to be preserved alongside with the original information and the original form. Now we will discuss the contents of the study plan, what they need to have. The first thing that they need to include is a unique ID, an identification of the study. So generally, the upper side of this study plans, we see this identification, the number of the study, it needs to have a title in which it is specified that different plaguicide that will be analyzed. Or as Javier mentioned this morning, we need to mention what the testing item is. In this case, is a plaguicide. We will be analyzing its metabolite. We need to uh, provide a justification and an objective of the study. Why will it be performed? And also the identification of the plaguicide. In this case, since it's a unique element, it will have a UPAC number or a CAS number. Identification of the tests, the purity that the reference and testing items must have, stability, and homogeneity.
Bueno, el segundo punto en el contenido del plan de estudio bastante importante es la información relativa
Now,
al
the second point in terms of the content of the study plan is that information regarding the sponsor and the test facility. This protocol, as I mentioned, it's to say who is the sponsor, where can you find this person, what their contact information for both the sponsor, for any testing facility, or any person interested in the protocol. They need to have the same information for the study director, who's the study director, where this person can be found, their telephone number, their email, because there can be any change, there can be any amendment, there can be any deviation, and need to have a point of contact that I can reach so that I can tell this study director what is going on with the study, or if things are going right, because this is also important. Also, the person responsible for the test, for both for field and laboratory. So this study plan needs to include who's the field researcher, their telephone number, their email, same thing for the laboratory researcher. And also who's the person in charge of quality assurance for all the inspections and the audits that will be performed. As I mentioned before, the study plan needs to include a series of dates that are very important. The first one will be the date when the study plan was signed. This is when the official kickoff date of the study. This needs to be approved by the study director and the sponsor, but it also needs to include the proposed dates for the beginning of the study that need to coincide with the approved dates, of course, and also the date for the end of the study. This will be included point 5 or 6 of the study plan. What are the testing methods that we will employ for the execution of the study as well? And there are some specific points, just as the justification of the study, the characterization of the test system, the testing system. You may remember from yesterday, our test system is the species in which we're working on. So in this case, cacao, pineapple, banana. And as we mentioned yesterday, we need to know the variety that we're working with. This needs to be a commercial variety. It needs to be representative as well. And everything that we discussed yesterday about the test system, the method of application, how this test substance will be applied, the study plan or protocol is not specific enough to say what the specific equipment that what is the specific equipment that will be used, because we could use different types of equipment. So for instance, if I'm going to have 4 tests in farm number 1, where I perform test 1, I'm going to use a certain motor equipment, But in form number 3, during test 3, I'm going to use a stationary pump. So in the same study, I can use different equipment. So the study plan doesn't go into that detail, that level of detail. Also, the doses and the frequency with which this will be applied and the duration. So let's say I'm going to apply it 4 times every 7 days, and I'll take samples on the 7th and the 14th day after applying the pesticide. This is something that is not included in our presentation. It's something that we have learned over time, and it's that these study plans need to be known by the administrator or the owner of the molecule, because they will be the person that will determine certain technical parameters in terms of the frequency and of the dose and the estimated time for taking the sample. So this is something that is agreed with the owners of the molecule, because they are the ones that have the technical information regarding its application. We need to have a series of records and reports. So in this study plans, it says 1920. The lab must have this document, or must keep this document. Same thing goes for tests, for any field tests.
Siguiente, por favor. Gracias.
Next.
Bueno, vamos a hablar un poco
Now let's talk a little bit about the implementation, the execution of the study. As I mentioned, the study needs to be an unique identification, which is this number that you see here. And there are some specific elements related to this number. For instance, it includes the year when the study started, the country where the study is taking place, and what the type of test is. So in this case, it tells us it's the 2012 318, which is the Spiritorani. This is a study to check the residue of the Spiritorani in pineapple. It is started in the year 2019. It's taking place in Colombia, and this is the study or test number 2. All the elements related to a particular study must bear this identification, so that we know what's the specific test that is taking place and where it comes from. For instance, in the case of the samples, the study samples must include a unique identification, which is the one that you see in point number 18. This ID of the samples typically come in an alphabetical system. So the samples A, D to D are typically untreated, and it tells us about the dose and the frequency as well. There are some specific tables for the processing of the samples. For instance, when we analyze different parts of the crop, either it's the pulp or the peel or the fruit as a whole. So there are specific tables for all of these. So when we take samples, for instance, in different time intervals, there will be a specifications, and there will be a table that will indicate us how to name the sample. So for instance, A and B samples are untreated. We have 12 fruits, and we will be analyzing the fruit without its crown. So the whole pineapple, but we have removed the upper part or the crown. And the CD are the ones that come from the second treatment or the treated parcel, and this we're taking 7 days after the last application. We will use 12 fruits, the whole fruit. So it is very important, and I would like to highlight this because it is necessary for you to take a sample and to know from which test it came, what is the identification of that test, and what type of sample it is, whether it is treated or untreated. Because for instance, you may have someone in your lab who helps you with cleaning the refrigerators, and this person may be cleaning where the samples are contained. This person needs to know that this cannot be defrosted and that they can mix samples A, B with C, D. So all of these things need to be taken into account.
Bueno, el estudio obviamente se debe desarrollar conforme al plan que
This
nosotros
study
ya registramos.
needs to follow the plan that was recorded, and we need also to record if there are any amendments, any deviations. We need to record all of these changes in an orderly way by the person who requires them. This is very important. This is data that needs to be clear, that needs to happen on site, that needs to be precise, and we need to be able to read it. This needs to be written by the person who is taking that information. Every piece of data needs to be signed and dated. In the lower part, there's a space that says signature of the study director and date. When we fill out this form, the study director needs to include their signature and the date, therefore certifying that all of the information contained above was issued by them, and it says that this information is true. This happens very often, and also with other quality systems whenever we have an error. So we need to make sure, for instance, I have an error here, a deviation of 1.811. Whenever I have these corrections, I need to cross, I cannot just cross this. I need to include, or I can draw a line. I will include the proper data and indicate what type of error. For instance, this is an input error. Why? Because I included the wrong number, but there can be writing, calculations, different types of mistakes. We have identified them. So for instance, in this case, is this EE is an entry error.
Los datos obtenidos directamente en forma de entradas informáticas, esto nos pasa mucho con los datos de
The
laboratorio.
data that comes from the lab, sometimes the data that is part of the software. So we must be very careful with this. Why? Well, first of all, when we don't have a configuration of the system, these can throw at us erroneous states. So we must be paying attention to the data that's being generated by the HPL systems. So first, they indicate the date where the sampling took place, and also the software should allow me to preserve the integrity of the data itself. Basically, that I could have a traceability of that crude data, that I cannot perform changes in that data without having control over it. we must indicate a person that indicated the change. For example, there was a formula in the software that was not what it was supposed to be. Well, if I do this change, the person doing it must sign, date, and even put the time at which the change was made. This is subject to the insurance quality group during the inspection process might revise the traceability and indicate the truthfulness of the data being collected, and also mention the reason why the change took place. Next. Very good. When in these type of study plans, protocol is very clear to see the number of applications that will take place. Generally, there is a tolerance of more or less 1 day. And also the dates in which the samples will be taken. So generally what we do is to have a schedule and we go to the field. And it turns out that on the field, the day that we're gonna apply the chemical the first time, then it rained all day. So all those changes that we have to the schedule must be reported by the study director. The study director is a person that must know in detail what is happening with the study on the field. When the study director, for example, as Ken, that is far away of studies that are taking place in other countries such as Panama, it is very important that anything should be informed, and anything and everything should be informed to the study director so they have knowledge and be able to make the necessary decisions when they need to be made. And among these, the schedule changes if it were necessary. We must complete in the form any type of deviation and send this as soon as possible to the study director, because he must value if the impact is high, medium, or low to the data. All the applications must be done on site and they must be sent to the study director as soon as possible. What this means is that the investigator will go, applies the chemical with the registration, and as much as possible in the same day or the day after, they must send this information to the study director so they know what was the efficiency of the application, if it was done correctly, or if it did not take place, for him to take the necessary decisions. Generally, applications have an acceptable percentage between 95 and 110%. So if there is some sort of deviation from these values, they need to know about this as soon as possible. Next. This is something that I mentioned before. If the staff cannot fulfill the implementation of the protocol, they must contact the director as soon as possible. So between them, they can discuss what the problem is and make the necessary decision. But it is the study director that makes the final decision, that approves whether or not the change or the amendment takes place. The amendments will only take place with modified instructions, and these will only take place if data is still being produced. If there's no data input afterwards, there's no reason to perform an amendment or a change to the protocol. And any deviation must be communicated to the study director. Next, please. This is something we spoke about. We need to deal with the deviations to fill out the information on the field data book. These deviations will be communicated through email, or even personally, which has been the case where the study director is in the application at the time of the application. But in the field book data, there is a part where we have communications. Within those communications, we must register that the field investigator was there with the study director present and what happened. This is the registration upon which we can have support of anything that happened during the time of the study or any decision that was done on site. We must include the reason why the deviation was produced, whether if it was a storm or rain, something completely unavoidable. It could be the case that close to the area they are applying pesticides with airplane application aerosols. And this must all be noted. And any type of deviation from the SOP should be communicated directly. My suggestion would be that the SOPs should be reading constantly because there is certain bias that I already know the SOPs, but the specificity is sometimes forgotten with time. So we need to look over these procedures over and over again to see if they're being done correctly or if they need any type of adjustment. Next, please. Great. Well, that is my presentation. I thank you all very much for your time. Eric, the floor is yours. Thank you for your time. Thank you, Edwin. As always, great presentation. Next, we have the fourth session for the day in charge of Saul Flores and Joe De Francesco, consultants, with the topic of the data field book. Saul, the floor is yours. Good morning, Eric. Please confirm if you can hear me, okay? Yes, we can hear you fine. Good morning to everyone. For those coming into the presentation today, my name is Saul Torres, and I will be continuing with the logical order in the presentation of the topics relating to good laboratory practices, continuing with the process itself, where a field investigator and the difficulties and challenges that they might have at the time of their work. Now we will talk about the application of everything that we've talked in previous presentations, referring to the filling out of the field notebook. For this application, we have 1 hour. Today's topic takes about 1 hour, and we will divide that into parts. As we get into detail, I will explain that there are certain sections in the field notebook. There is one section in particular, which is the most extensive, and this will be explained by Joe. After Joe is done with this part of the explanation, then I will come back and explain the end or the rest of the sections on the field notebook. Next, please. Very well. The field notebook is the basic file, the indispensable file that is filled out when we develop a field trial. As the rest of the documents that you have heard about these past days, it has its components independently of the trial that's taking place. We always have common elements in each one of these field notebooks. Basically, these are 9 sections, and when I say a field notebook, I refer to an investigator or the local coordinator will receive the formats in this in a Word file in Spanish and English, so every field researcher can fill these out according to the activities taking place. We do not need to elaborate these. These are just empty templates that are filled out. The field notebook has 9 sections, and they also follow a logical order of how the activity or the trial will take place. If you could go back. First, we have a statement of good laboratory practices, staff registration, communication, test substance records, trial site information, applications, sample harvest, sample shipping, and materiological. Next. Adriana, Veronica, can you confirm that we lost Saul? Yes, my screen is frozen. Yes, we will give him a moment to log back in. Excuse me. I'm sorry, I'm back. Issues with the internet connection. Can you hear me? Yes, we can hear you fine, and we can see you as well. Don't worry about it. We know that this is absolutely normal, and you may continue. Thank you. Yes, next, please. Very well. As Edwin explained, these templates are filled out by the field investigator. They must be filled out by hand. Here we forget about automatization. They must be filled out clearly with good handwriting, and all the information is filled when the event takes place. It is the case that when we begin with applications, the applications finish, and immediately afterwards, the field investigator sits down in the plot in the same table that they used to do the different calibrations, and they start filling out these forms because we cannot leave things to our memory. Possibly our plots are close to the office, and maybe we would like to be comfortable in our office, but in that last lapse of time, some information might be lost, and that may compromise the fidelity or good laboratory practices for the study. Next, please. Next. Very well, as Edwin indicated, every trial, every project has a number, and every trial has a ID number. This is the example that we have here in Ecuador. The ID, the project, and we belong to project 12-318. We put the year that we began, the initials of the country, and the trial number that we're doing. So in this case, our first page in that field notebook shows this information. This is the trial that took place last year, and that we will begin next week with a new one with a new number, but it will be 02. Instead of EC01, it'll be EC02 with the second trial. Well, this explanation is also printed out in the field notebook given to every field investigator, researcher, and it has all the information that Edwin mentioned in the previous presentation. These are general aspects of how these should be filled out. Nothing is deleted. If we need to make a correction, then we correct, stating that the correction took place. There's no blank spaces. What does not apply, or whatever is not applicable, must have an end inclined line filled, meaning that it's not filled out, and every error or any correction that takes place must have a category according to the type of amendment or a correction. We do not write in the back of the pages. There's only one face to each one of the sheets of paper, and that's all we use. Next. Very well. This is an example of our field notebook, and here you can see in the bottom of the screen, we had the intent of inputting data of 2,240 milliliters. Nonetheless, we had an error at the time of writing, and we noticed at the time this was written down. So what the field researcher does is to cross out the incorrect data, put the correct set of data, and then we put a code of the error. In this case, it was an input error. That's what EE means. Next, we have the initials of the person doing the amendment and then the date that it was corrected. Next, please. Very well. Getting into the topic at hand, and here I want to show you something. Next to me, I have our field notebook, and this field notebook I have this here at the time being because right now we are in the facilities in our investigator office. So this field notebook is in complete custody of the researcher until the time of the investigation ends. This document does not travel with the person. I made use of the circumstance of being in this training session because precisely now we're working with our researcher to organize the application for the second trial. So we decided to do the induction from Santo Domingo, the province where we perform our pineapple studies, and show you what this field notebook looks like. And this information is under the custody of the field researcher and is filled out as the trial goes by. So the first part is the fulfillment of good laboratory practices, and this is filled out when all the document is complete, and it's a statement in which we indicate that we have fulfilled good laboratory practices and procedures, and it is signed by the field researcher. As Edwin explained in the previous presentation, here also in the field notebook, we must have a registry of the deviation scenarios in the protocol. As we saw in detail before with Edwin, we try to be very thorough to what the protocol says in our study plan, but there can always be matters present that take us out of the framework and the conditions that are required by the protocol. I will just tell you an anecdote that of something that happened to us. We had a deviation of the protocol, but deviation did not happen on the field. It was a deviation that took place when we reduced the samples. What happened is that the protocol specified a maximum weight after we already had reduced the samples, and it turned out that since our pineapples were a little bit bigger than expected, but these are the conditions under which they are harvested, the end weight was quite higher. So we had the need of write down a deviation to the protocol. This is something that falls out of our hands, but it's something that can happen on the field. Next, please. Next. Yes, we covered this. If you could go to the next slide, please. Next. Well, the second part of the field notebook is the staff or personnel form. This is where you fill out the information of the people participating in the trial. The greatest importance, as Edwin mentioned, is the field researcher itself, but nonetheless, we need to register all the people participating. It is important to annex the CV of the person that's printed out, also the statement of confidentiality, the designation of the person, and also important to have the training log, especially in this, which is as specific as good laboratory practices for each one of the people. There, we need to have an annex of the training they received. Next. Next, please. In the part of communication that Edwin also mentioned, we must fill out by hand the registry of communications. What I mean by this is when we, for example, finish applying the substance or the chemical, we fill out in the book and we scan and remit this to the study director, writing down in this registry the date, the initials of the person generating the communication, and the observation of what that communication entailed. For example, email on January 2nd, we mentioned from this person to that other person, the application of the different procedures. It is necessary in the majority of the cases to print out these emails and annex them once the field notebook is complete. Next. Now we will see the substance, the trial substance. Next, please. Very well. The trial substance. Before we begin working on the field, we must already have in our possession the substance. And here we show in the book how this substance came to us. It could be a kilogram, grams, liquid, solid, different weights. And always we must give a description of the containers, the temperature, and maybe next you can see some presentations, some pictures that we have. Next. Great. Well, here you can see the form that is filled out. On the left, you see the way in which we received the test sample. This came by air mail, and we need to make sure that to state the conditions, how it arrived in our hands. What you see in the bottom is what one of the most used pieces of equipment in these trials, which are data loggers, which are thermometers that we can put with whatever time frame we want for temperature registration. And the substance from the beginning must have a temperature registration, because we also must state that the test sample has been stored at the adequate temperature, so there's no degradation, and the exact the validity of the data stays intact. Next, please. Up to now, all the registration procedures we have explained, some have annexed documents. In this case with the substance, there's also information that is included in the package where we receive the substance with a analysis certification and labels in case that it is employed. The invoice and the shipping method to make sure that we're using the substance that was sent from the owner of the molecule itself. Next. Very well. Now we will discuss the area corresponding to the information on the test site itself. And as we have mentioned before, the end of all this data and file handling and all these filled out forms is to be able to be very clear when a person that is not involved in the trial itself to be able to understand what we did, for them to be able to make sure and have certainty, evidence that all the experimentation that took place. And this is why it is so important to have all the information that may be generated even before beginning the application process. Up to now, all the information that we have seen can take place before the application on site itself. So the information and the trial site is 1/5. It's basically an X-ray of this notebook and the plot of land where we will be working. And this is filled out with information corresponding to the address, the idea of how you get there, the planning, information with the soil, a soil analysis. That's also an annex document, the history of everything that has happened in that plot, the cultural practices, applications of previous pesticides, fertilizers. And we must take into account that these plots, I'm sorry, there's an open microphone. The plots, if well, it is true that we have plots receive conventional treatment of nutrition and sanitary control that the owner of the land or the farmer has. We do not exclude everything. If we need to fertilize, if we need to control a pest, then we do so. But these registrations, all these previous treatments, so if you could stop for a moment, we started listening to Spanish channel interpretation and French. So please, if the interpreters, please go back to your channels. I hope this works.
Okay. I hope that this has been corrected. So please continue and we will verify that everything is working as it should. Next slide, please. So can you go to the next one, please? So these are some examples of the directions. We need to draw a map that indicates how to get to the plot. Next one. The apps that we may have on our cell phones, such as GPS, will help a lot so that we have this, we can have the specific coordinates and the directions and how to get to the plot. Here we see the application of a very simple tool. We have a diagram of how to get to the plot so that any person who audits or checks the field notebook can confirm how to get to the site. We need to, once we have the plot information, we need to have a drawing or a plan as detailed as possible, showing where the points of reference, where they're located, if we have a stream, a forest, a path, the distance from the untreated to the treated plot. It doesn't have to be in scale, but the information that is drawn needs to be as close to the reality as possible, so that any person who's not close to the trial can have the proper directions and know where it took place. Next.
Oh,
Yeah. Gracias, gracias. Ahorita vamos a explicar la parte 6, que básicamente se llena
Now we
con
will
cada una de las aplicaciones.
explain the sixth point.
Esta es la parte que
These are the application records, and Joe will be in charge of this part. Joe, good morning, and now I give the floor to you.
good. Thank you, Saul. And good morning, everyone. Good afternoon to our participants in Africa. As Saul said, I will be covering section 6 of the notebook. There are 9 sections. Saul covered the first 5 adequately. And as he mentioned earlier, section 6 is really where the information about the application itself gets recorded. And we tend to think of this section as very important because this is where there's information about the application equipment. You will, and we'll go into these topics one by one, but in general, section 6 talks about and asks for information about the application equipment. It asks for a photograph or a diagram of that equipment, so anyone reviewing the notebook can have a better idea of what it looks like. And very important in this section are the calculations that the field research investigator is using to determine exactly how much test substance and how much water is needed to apply to the plot to meet the protocol requirement. Excuse me. There's also information requested about the sprayer or the application equipment, the discharge calibration. We want to know how much the equipment is discharging per second. There's also information about speed calibration, how fast you are moving through the plot. And then when it's time for the actual application, the field research investigator will record the exact amounts of test substance and water that was, oh, I'm sorry, I heard a speaker on. That there, you will record exactly what's been put into the spray tank as far as the amount of test substance and the amount of water, the time of day that you made the application. We're going to go over these in more detail. There's also a form that prompts for information about weather at the time of application. Information about the status of the crop, how vigorous it is, whether it's healthy or whether there are problems. And then the recording of the actual pastimes, the amount of time that you actually spray the plot. There's a target time that you calculated on paper. And then when you're in the field, we want to know exactly how much time it took to spray that plot. And then at the very last, information about rainfall and irrigation. So now we're going to go through each one of these forms in section 6. Next slide, please. Thank you. And I do want to point out that all the forms in the notebook are there to prompt you to provide the information that is necessary and needed to verify that the data is valid. The forms are specific to different kinds of trials, but also I wanted to point out that they are suggestions. If you decide that you want to provide the information in a different format, you are very welcome to do so. But the information needs to be recorded in the field data notebook. And we tend to think that the forms that we've created just make it easier to get that information. But by no means do you have to use these forms. If you decide you want to use a different format or a different form, that's fine, as long as the information that's on the original form is also included in your particular information sheet. So the first form in there is about the spray equipment. And this is where you provide information about the sprayer itself. Are you using a tractor? Is it a backpack sprayer? Or maybe in some cases, it's not a spray at all, and it's a granular application. So as you can see, this is indicated on the form. We want to know the type of propellant is used in a sprayer, the pressure that's used, the number of nozzles, etc. Anything to do with the spray equipment gets recorded on this form. Next slide, please. And then there's a blank sheet here asking for a diagram or a photo of the application equipment. So I'm going to give you a couple of examples next, but if you are using a boom sprayer, which has one or more nozzles, if it has more than one nozzle, you need to indicate on that diagram or in the photo the number of the nozzle. You need to number them because when you do the calibration, we want to also know the output of each individual nozzle on that boom. So if you have a 10 nozzle boom sprayer, you will indicate nozzles 1 through 10. And when you record the output of those nozzles, they will correspond to the exact nozzle on the boom. Next slide will show examples of different types of application diagrams and equipment. Next, please. Thank you. So here's 3 examples. The upper right is a photograph of a 10 boom sprayer, and you can see that the spray system is indicated as sprayer number 4. The boom that's used with that sprayer is boom number 8, and then the individual nozzles are identified as 1 through 10. On the left side is a combination photograph and a sketch, and it shows the researcher holding the 3 nozzle boom, and again, the nozzles are indicated 1, 2, 3. It's spray system number 1. It's a CO2 carbon dioxide tank, and it's put into a backpack sprayer. So he will put that on his back and apply it vertically to the raspberry plant. And then on the bottom left is a very crude drawing of the equipment. And again, this will suffice because it shows the sprayer, it shows the tank, which in this case is a 3-liter bottle, and then it shows the 3 nozzle boom and where the nozzles are on that boom. Next slide, please. One of the GLP requirements is that you keep a log of your instruments and equipment that's used in the trial. And this is a form that can be used to record that. Any repairs that are made to equipment, any maintenance that's performed gets recorded on this form. And it indicates to a reviewer that the spray equipment was checked out for appropriateness to the trial and that it's working properly. So in the beginning, before your first application, you'll want to run the sprayer with water to make sure there are no leaks, no cracks in any of the hoses, and you would record that. You would just indicate the date and your initials that you did perform maintenance and that everything looks good. And then during the trial, if there happens to be a problem with the equipment and you need to repair it, we want to know about that. And again, this form can be used to record any repairs that are being made to the equipment. Next, please. This is a very critical form. It's mostly blank, and this is where you indicate the calibration, the calculation, excuse me, the calculation of how much test substance and how much water needs to be applied to the plot to meet the protocol requirements. As Edwin and Saul mentioned, the protocol is a recipe that you follow to use the right dosage, but that's indicated in usually milliliters per hectare or liters per hectare, but your plots are going to be smaller than that. So you need to calculate how much test substance and how much water will be applied to your specific small treated plot. So this is a very critical calculation, and it is based on your particular plot size and the protocol. And so the area up above, which shows the formulas that you're going to use, that way your reviewer will know exactly how you came up with these numbers. So basically I see it's in Spanish, but basically it's the area treated is the length of the plot by the width of the plot divided by the hectareage, which is 10,000 square meters per hectare. The protocol indicates that you want to apply 500 milliliters of test substance per hectare. And usually the protocol will give a range of water that can be used. And in this case, the researcher chose 700 liters of water per hectare. And then the bottom of that form is where you actually show your calculations with specific information to your particular plot. In this case, the plot is 30, and these are made up numbers. The plot is 30 meters long by 10 meters wide, which is 300 meters square divided by 10,000 square meters per hectare. So your particular plot is 0.03 hectares. You know that you want to apply 500 milliliters per hectare. You multiply that by the plot size, and this indicates that you will need 15 milliliters of the test substance to be applied to your particular plot. I think there's a typo error there, but the researcher decided to use 700 liters of water per hectare times the size of the plot, which is 0.03 hectares. And that indicates that there'll be 12 liters applied to that particular plot. So basically, those are critical calculations that if you apply 15 mils of test substance in 12,000 milliliters of water to your plot, you will achieve the protocol rate. Next slide, please. It is in Spanish, so I'm going to have to look at my English slide. And Adriana, I don't know if you want to switch over to English slides, or should I, maybe it's just one slide, I'm not sure. Oh, can't see English. For some reason, I'm seeing Spanish. But if all the English speakers can see English, I have a slide on my other computer I can look at because I can't read the Spanish. So this form is a form that puts the calculation. Yes.
Joe, podemos hacer una pequeña prueba en su computador, en la pantalla.
Can we test?
Oh, somehow I switched over to
view
there
options.
We go.
You can choose English, the English presentation.
Yeah, for some reason it switched over to Spanish. I've got it now. I can see English. I hope everyone else can. I'll continue if all the English speakers are on the correct slide. So this form is showing what we call overage, or a calculation to determine how much extra you want to put in your spray tank. What we calculated on the previous slide was the exact amount of test substance and water that you want to apply to the plot, but you don't want to measure out and put into your tank the exact amount, because there's going to be some of that solution that needs to go through the system and fill up the lines. There's going to be, you need extra solution so you don't run out of solution by the time you get to the end of the plot. So you always mix up extra, and this is what we call overage. And you can see circled in red, it says that you can choose any amount of extra that you want to put in your tank. Some people like to add enough extra so the numbers come out even, so they're not measuring small amounts. They add enough to fill up their tank. So it's whatever you want, but you always need to have a little more solution than what's called for in your original calculation. And then here's an example. We know we wanted to apply 15 mils per plot. So in this case, the researcher is going to mix up 20% more. So that means that he's going to add 18 milliliters of test substance to the tank. Same percentage is used for the water. Instead of 12,000 mils, he's going to add 20% more, which brings it up to 14,000 mils. So now that researcher will add 18 mils of test substance in 14 mils of water to the spray tank. The bottom part of this form helps you determine how long you need to spray that plot to get the correct amount to the plot. So we know we wanted to deliver 12,000 millimeters. And in another part of this section, there'll be a calculation or where you calibrate your sprayer. And so I took that number, and my sprayer, or the sprayer in this case, is discharging 57.6 milliliters per second. So it's a basic calculation. We want to deliver 12,000 milliliters of solution. The sprayer is outputting 57.6 milliliters per second. That means I need to spray the plot for 208 milliliters. And if I do, I'm sorry, 208 seconds. And if I spray for 208 seconds, then that should be the correct amount to deliver the protocol rate. In this case, there needs to be 2 passes through the plot. And so you divide the total seconds by 2, and that means pass number 1 will be 104 seconds, and the second pass will be 104 seconds. If you needed to make 4 passes, you would divide 208 seconds by 4, and that would determine how many seconds you need to spray per plot. So again, these are, the forms are there as prompts for you, but we would like to see the calculations. That way we can verify that the correct amount was being mixed and sprayed. Next slide, please. Okay, this is the form for calibrating your sprayer. And this particular form is for a boom sprayer. We do have other forms, different forms, if you're using a mist blower sprayer, which does not have individual nozzles that you can collect the discharge. In this case, this is a boom sprayer with several different nozzles. And so in the protocol, it will indicate that to be valid, you need to do 3 runs of output. And then you would take the average of those 3 runs, and that would be your sprayer output. And that's the calculation you would use in the previous form that I showed you. So to be a valid calibration, it must be an average of 3 runs. And this form prompts you for information about the sprayer again. And then at the bottom is where you actually write down the information as you're calibrating your sprayer for each run. On the left side is run number 1. You will have a stopwatch to indicate the time. You usually want to do it for at least 30 seconds. And then however many nozzles you have, you stop your sprayer, and then you measure the output from each individual nozzle. And you do this 3 times. And then there's a calculation at the bottom. You will get your average output divided by your average time, and that will be your discharge rate for the sprayer. There is a requirement in section 14.2 of the protocol that indicates that you need to make sure that each nozzle is spraying within 5% of the average, and that each run is within 5% of the average. So very carefully, you would calculate, for example, to determine whether nozzles are being consistent. In other words, you don't want one nozzle be delivering 300 mils per second and the second nozzle to be delivering 400 mils per second. It just creates an uneven spray in the field. So you want the nozzles to be sprayed within 5% of the average, and that ensures consistency. I won't go into the details of how to do that. I do have a couple of examples here, but it becomes self-evident. And if you have any questions, of course, you could always ask someone like me, a consultant, or the study director for the project. Next slide, please. And this is what's called a recheck for the boom sprayer. So if you have multiple applications for a particular trial, in some cases, the protocol indicates that you need to make an application every week, every 7 days, 4 times, and then harvest the sample. So for multiple applications, if the parameters of the spray equipment has not changed, if the settings have not changed, or any other parameters have not changed, then you can save time by doing just a recheck, and that's just doing it once out in the field. And it's just really to save time. If you want to do a 3-run, you can, but the 1 run, you collect the information there and you compare it to the original 3-run calibration. And if it's within 5% of the original 3-run calibration, then that means your sprayer is still performing according to how you originally calibrated it, and you can use the spray equipment and spray with confidence. But if you decide to do a new 3-run calibration, or if that recheck is not within 5% of the original, then you must do another 3-run calibration. And you would just make a copy of the 3-run calibration form and make a new 3-run calibration. And then it's important to remember that from there on going forward, it is that 3-run calibration that will be used in all your calculations. So the most recent 3-run calibration is what is used in all your calculations to verify whether you've reached your target rate of the protocol. But if your single runs are within 5% of your original 3-run, then you don't need to do another 3-run calibration. So it's up to you as a researcher to decide whether you want to do a new 3-run every time, or whether you want to just do a recheck in the field. And that's what this form is for. Thank you. There's also forms for this calibrating your speed. And there's on the left side of the screen is the 3-run speed calibration. And then again, like the sprayer calibration, if you want to do a recheck for your subsequent applications, you could just do a 1-run just to make sure you're in the target range for your speed. Next slide, please. This is the form that's used for on the day of application for all the information about the sprayer and what you're putting into the sprayer. In this example, there's a column for 2 different treatments. In most cases, your trial will only have 1 treatment, so you can line out that second column. We've had trials where there are 2 treatments for every application, and that's why there's a form that allows for 2 treatments. But usually it's just 1 treatment, 1 treated plot, which is treatment 02. But this form is asking you, they just want to make sure that you have the right test substance. So you write down the test substance, the lot number that came with the certificate of analysis. We want to know when you mixed the water and the test substance and when you applied it. And this is so we know that you did not mix at 8 o'clock in the morning and spray at 4 o'clock in the afternoon. Sometimes a pesticide will degrade in water over time, or they'll separate. So we want to know the time interval between the time you mixed and the time you actually applied. And then the rest of the form is asking exactly what equipment did you use, how much water did you put in the tank, how much test substance, et cetera, et cetera. So this is filled out on site at the time of the application. Next slide, please. This is additional information. And again, this is filled out in the field. And it's asking for conditions at the time of application. It's asking about the crop. Is it healthy? How high is it? And this is important in a vegetable crop where it's an annual vegetable crop where it's growing as the season progresses. So we want to know the crop height. What stage of growth is it in? Is it vegetative? Is it flowering? Is it fruiting? Whether there's moisture on the surface, the wind speed, the air temperature, again, environmental conditions during the application. This is all very important. Form props for the crop status and the weather parameters at the time of application. Next slide, please. This is to write down your actual pastimes, and that's the top part of the form there in the box. This form allows for 4 passes, but if there are more, we can create a form that allows for more passes, or you can write included in the notebook. But this is the actual time that's being recorded. Who is recording the time? The discharge rate of the sprayer, which you've gotten from the other form when you did your calibration. Now that you know the time, the bottom part of that form is a calculation to verify apply the correct amount of test substance and water. And the protocol allows you to be within a range of 5% under the protocol rate, and it should actually be 110%, 10% over the protocol rate. So you have a range of 95 to 100 rate. And the calculations form that we put in here with the blanks is something that we put together to help prompt you to write down and show your calculations, but by no means do you have to use this particular form. You could do it on a separate piece of paper in your own words. Information is there. We want to know at the end of the calculation that you are within the protocol rate, within 95 and 110%. And we want to know that you've met the protocol water requirement also, that you have not applied too much water or too little water. So that's what this form is all about. And this is a very critical form in the notebook because it shows us exactly how long you spray the plot and whether you've met the protocol rate. And if you have not, That's okay. Sometimes it happens. Sometimes it takes a little longer to walk through the plot for some reason. So more test substance has been applied. But if it's not between 95 and 110% of the protocol rate, then you need to contact the study director and tell him or her that you have a deviation, and he or she will decide whether it's a critical deviation and will indicate how you should proceed. So this form is very important, and the study director will want to know if you've met the protocol rate for every application. So it is important to notify your study director. Can I have the next slide, please? This is the last form in section 6, and this can be filled out after each application, but it's to indicate when a rain occurred, if it occurred at all, and when irrigation occurred. And this is to let the reviewers know whether perchance that it rained immediately after your application and maybe the pesticide got washed off, which would not be good. So this kind of information, if you have records of rainfall in your particular area for where the plot is located, we want to know how much rain fell and how much longer after that application did it come. Was it days later or just hours later? Same thing with irrigation. If there is irrigation, be it overhead irrigation, furrow irrigation, or drip irrigation, we want to know when the plots were irrigated. And then as you go out to the plot for subsequent applications, it's good to look at the treated plot just to make sure there is no phytotoxicity. In the bottom part of the form is a spot where you can record any phytotoxicity that you may have seen. So if you go out to your plot every 2 weeks to make an application, before you make that application, check the plot, the treated plot, compare it to the untreated plot to see if there's any toxicity that may have occurred from that pesticide treatment. We will want to know that, and so will the owner of the molecule, the registrant of that product. So this form allows you to indicate whether there's any phytotoxicity or not. So those are all the forms that you will find in section 6 of the notebook. And as I mentioned earlier, you can use your own forms as long as the data that we're asking for is recorded somewhere in the notebook. As indicated by Eric in the beginning, we will take questions at the end of today's session. So thank you for your attention. And I will turn it back over to Saul, who will cover the rest of the field data notebook. Thank you.
Muchas gracias, Joe. Efectivamente, estamos, ya hemos
Thank
visto.
you, Joe. Yes, as a matter of fact, we have seen the first part, which is the statement of good laboratory practices, this part that has to do with the staff, the field staff, the field notebook information from the plot. And Joe just explained how this data is filled out. And the next logical order is to fill out the information corresponding to the harvest. As you may see, any protocol or any study plan has a specific date in which we must fulfill these activities after the last application. Having passed some time, we have the harvest of the sample. And what we fill out as field researchers is basic information that might sound obvious, but must be registered with respect to how the harvest took place. So we must have a statement saying that we have followed the corresponding order with not taking, for example, fruit from the borders of the plot if we used different set of staff, the time that it took place, and also a registration of the supplies, the inventory, how many samples we're taking. Please go back. How many samples we're taking. And in the same way, very important with respect to the harvest and storage for transportation or shipping of these samples to the lab, we must have a temperature control. Simply to state that the harvested samples on the field were transported adequately in conditions as far away as possible from high temperatures. Also, in this part, we have a statement in this document that avoids cross-contamination with use of equipment. There must be an idea of when these plots are being treated and all the other details entailed in the procedure that has to do with the harvest. Next, please.
Entregar el producto que ha
Delivering the product or the result obtained from the field researcher by harvesting these samples to be sent to the labs. The final analysis of these samples are done after the labs have gone through a classification and data and sampling reduction. And what you see on the screen is an example of how the information must be filled out, where we have specific data in the way that it was transported, if it was a refrigerated truck, if it was air shipping, when the samples were taking, the temperature, etc. As I said in the beginning, managing this field notebook also has to do with annex documents. In this case, the data lodgers that goes in each one of the coolers with the cutting samples. This behavior, this temperature behavior from the time they leave the field and get to the lab is also part of the annex document. Next, please. Very important to have traceability. We must show that there was a chain of custody. In this case, if the field researcher stays in the plot of land after the harvest to destroy the rest of the fruit and delivers this to someone else, for example, a local coordinator for the transport of the sample, there must be a registry of the passing of these samples from one hand to the next, and the person that has custody and the transportation of the samples also must register when the staff receives the samples. And as I said, there must be a temperature registration. Please go forward. Next. Yes, this is the verification list of a sample, of the samples going to the lab. This format may be managed by the staff in the lab receiving the sample, where it states in the way that the sample arrived, the conditions, the temperature, and if everything went according to what the protocol says. Next, please. The last part of the field notebook is something that, if we leave it at the end, is registered throughout and even before the first application, and it has to do with the meteorological data. Depending on the case and the conditions and the protocol, or what the director of the study states, we must have registration of the meteorological conditions 1 month before, or a set time before, during the application or the development of the trial, and even after the samples are harvested. There are certain times depending on each condition and each trial, and this information also must be annexed or added to the field notebook. We have passed through the time for this information, but we end with this processing of the field notebook. We sign the first sheet, which is the statement of good practices, and at that time, we start numbering all the pages of this field notebook when we have all the information already compiled. After this, the field notebook will have to go through the necessary channels for its storage, and therefore it's necessary for the audit process when they deem it worthy. With this last slide, we have covered the hour that we have assigned. If it's true, it's a lot of information. We have tried to give you some general aspects and the most important details, but we learn definitely working with this field notebook when we need to do it ourselves. The message here is that everything needs to be registered. It's not forbidden to make mistakes, but it's a learning process. And this is basically what we had to say this morning, and we hope that this helps you going on forward for when you yourselves have to work with your own field notebooks. Thank you very much. Goes back to you. Thank you, Saul, consultant of Agrocalidad Ecuador, and Joe DeFrancesco, consultant of the foundation. We have had from yesterday some very long sessions, and we have heard in the chat on the phone a recommendation that I think is quite appropriate. These are 4-hour sessions, and at this time, we'll have a very brief pause to have a group picture. Yes, the group picture. Yes, forgive me, Eric, for interrupting. We can barely hear you. I don't know if maybe the microphone. Oh, now you can hear me. Yes, now you can hear me. Yeah, I had the microphone in the wrong position. I was mentioning that these 4-hour sessions are very long, so I had a recommendation over the phone that we will implement. The group picture is taken always at the end. We will take it at this time to have a sort of pause or relaxing activity. And all those that can connect their camera, I would ask that you do so. There's a lot of people. We have a lot of people connected right now. So I am sure we will have a lot of screenshots. And I would ask my colleagues to please help us, Veronica, Adriana, Maria. Maybe you could all help me to take screenshots of all the people to have a very good group photo. There are people from Latin America, we have people from the Caribbean, we have people from Africa, and therefore it will be a picture that will show great participation of this event. I hope that my colleagues are taking the pictures now, the necessary pictures. Please forgive me for the interruption. Just one indication. Zoom tries to accommodate at the beginning the people that are turning on their cameras. So leave your cameras on till the end, because it'll start moving people around and it changes the screen. So we will let you know when you can turn off your cameras, those that wish to turn on the cameras for the group photo. Great. At least on the screen that I am seeing here, I see people from Uganda, Senegal, Colombia, Argentina. This is a very representative picture. We have a lot of people interested on a topic and united by the efforts of the foundation. I see that my colleagues tell me that people have been taking the pictures, so therefore we will come back after this relaxing moment to the presentations. I thank you all very much for connecting your cameras. Before we go with Ken, I will do the same thing I always do. I want to remind you the 2 key buttons we have here. I've said this many times, but it's very important. This is very important. In the bottom, you have the interpretation button. It's that little world icon. If you click on that, you can pick the language where you want to hear the spoken. And the top part, you won't see this right now because nobody's sharing the screen, but on the view option button at the top, you can select the language in which you want to see the PowerPoint presentation. And these 2 buttons are key, and we hope that all of us have become expert by this time in the use of this platform. So we will continue with today's presentations. We have another presentation, one very important, that's 1 hour long. This is about the report of the study. Ken Samuel, he's a consultant. We had him yesterday with one of his presentations, and we have him again today. Ken, thank you very much for these great presentations, and the floor is yours.
Thank you. Hello, everyone. I will be presenting my talk in English, and I prepared it with input from Joe DiFrancesco. Next slide, please. A final report is required for each GLP study that has been completed, and a study consists of all of the trials conducted under one protocol. So, for example, I am the study director for 2 separate studies with indoxacarb on coffee. One is being conducted in Costa Rica and the other in Uganda. Each has their own protocol. So when those studies are completed, I will be preparing a final report for each one separately, even though they will both then be submitted to JMPR and subsequently to Codex in order to get an MRL established for indoxacarb on coffee. The report may be written to meet the requirements of a national regulatory agency. In my country, that would be the US Environmental Protection Agency. And then that report is suitable to be later submitted to JMPR and Codex. Alternatively, it could be written only for submission to JMPR and Codex and be written to meet the requirements of OECD. I have not yet seen a report that was written that way. I think it is probably easier for the writers to follow the guidelines of the country they're working in, rather than create a separate template. Next slide, please. The draft report may be prepared by the study director, or it may be prepared by some other person who has been hired to write the report. But the study director and the sponsor are ultimately responsible for the contents of the final report. Next slide, please. If someone else has prepared the first draft, the study director reviews the draft and makes any necessary revisions. The report is then submitted to a quality assurance reviewer to be audited. The study director then responds to any findings that the auditor has made and revises the draft report as needed. The study director's management, typically the study sponsor, must approve the study director's responses. After a quality assurance statement has been received for insertion into the report, the study director and the sponsor sign the report. Next slide. OECD has specified the contents of the report, and this is quite similar to what is required by the US Environmental Protection Agency. So I think this will be correct for just about any country in which you may be preparing a final report. So to begin with, the report must have a title, something that identifies the study that is discussed within. Identification of the test item, or the test substance, the pesticide that is applied to the crop. Identification of the reference item. So that is the more pure chemical that is the active ingredient in the pesticide. Characterization of the test item. That has been discussed previously, meaning the tests that are conducted to show the purity of the test substance and also the reference substance. So if the test item is supposed to be 30% active, the characterization might show that it is 29.7% active, and that is still very close, that is acceptable. And there would be a certificate of analysis in the final report that would show that. The reference substance is perhaps expected to be 99.9% of pure, and it may be 99.87% or something close. But again, there would be a certificate of analysis for the reference substance. Next slide, please. The sponsor's name and address must be in the report. So I'm now working with the Minor Use Foundation, and that has an address based in the United States, and so that would go into the report. the name and address of the test facilities or sites. So the places where the field trials actually occur and the analytical laboratories that do the residue analysis. The study director's name and address. And the address, since I am working with the Minor Use Foundation, when I prepare a report, I'm going to use the foundation's address. But a study director in another country who is affiliated with another agency, another organization, should use that organization's address. The field research investigators and laboratory research investigators' names and addresses. Now, those addresses are probably going to be the same as the test facilities, but conceivably the field research investigator could have an office at one location and conduct the trial at another location. So that needs to be clear in the final report. Contributing scientists' name and addresses. There aren't often contributing scientists providing reports to go into these magnitude of residue studies. But if there were a sort of a separate sub-report for some portion of the study, it could be a processing report that needs to be included. Next slide, please. Okay, the experimental start and completion dates. The experimental start date is the date that the first application of the test item was made in any of the trials within the study. So if there were 2 or more trials and the earliest trial had the first application on June 1st, then June 1st of 2022 would be the experimental start date. The experimental completion date is the date that the last data is collected in the analytical laboratory. So it's when it can be when the last treated sample is analyzed in the lab, when they receive the results of that sample. If they have to do a freezer storage stability study, that is part of this overall study to show that the samples, that the residues remain stable in freezer storage, then that perhaps would be the last data collected at the lab. And in order to determine that, if it is not clearly stated in the analytical report, then the report writer just needs to go through the analytical data and determine what the last date is that is mentioned. The report must include a quality assurance statement that is received from the auditor, listing the types and dates of inspections that have been made, the phases inspected, the dates that results were reported to management, to the study director, and to the field research investigator or laboratory research investigator as appropriate. And this statement also serves to confirm that the final report reflects the raw data. So if you are conducting trials and you're having audits on your data or on a critical phase, your standard operating procedures might not require that the study director sign off on the audit, but it is still required that you provide the study director with copies of those audits and your responses. One, because the study director needs to be able to approve also that everything is okay, but also because this information is needed to go into the quality assurance statement. The report must have a description of materials and methods. What is it? What is it that is, how is the test substance supposed to be applied? And just a basic description of what the protocol is asking for. There needs to be a reference to the guideline, the test guideline that is used. I wrote it as, in the slide, as reference to OECD test guideline, but actually if you are writing a report in compliance with, say, the United States Environmental Protection Agency guideline, that would be called OPPTS Series 860 guidelines. That's the name of the guidelines used in the United States. There's a different test guideline in Canada. Each country may have its own separate guideline. But alternatively, I believe it would be OECD 509 crop field trial guidelines. But as I said, I have not yet seen a report that references that particular guideline. Next slide, please. Then there is a summary and presentation of results, which needs to include all of the information that is required by the protocol. So were the applications made at the specified intervals? Were the samples collected at the dates needed? Was any kind of sample reduction required, cutting into quarters, perhaps separating peel and flesh? That needs to be discussed in the summary. And then also a summary of the analytical work, the method that was used, a very brief, 1 or 2 sentences, perhaps, description of the method, and then the results obtained in the laboratory. And then an evaluation of those results and conclusions. Typically, a conclusion would just be that the data presented in this report can be used to support a maximum residue limit proposal. The location of where the protocol, samples of test and reference items, raw data, and final report are stored. So basically, that's called the location of raw data. And there are often several different places depending on which of these we're talking about. So the protocol, if it's coming from the Minor Use Foundation in the United States, maybe, and the final report may be stored, may be archived here in the United States. Samples of the test and reference items should be maintained by the registrant who supplied them. So there'll be a separate address from that, which ideally is indicated on the certificates of analysis, but sometimes follow-up is needed with the registrant to confirm that. The raw data for the field is probably archived at your field trial location at the location indicated in the protocol, and the laboratory data will be archived by the laboratory. So there could be half a dozen different archive locations for these items. Next, please. The final report does not include copies of the entire field data notebooks. That would be far too many pages for the reviewers to have to look at. The field trials instead are summarized in the text of the report, and then there are tables included in the report that have the most important data entered into the tables. So the application rates are entered, the spray volumes. The tables will include information about the plot size and the crop variety. The maintenance chemicals that are applied to the plots should be listed. And then some information that would help a reviewer determine that the applications were done correctly. So the pass times, the single total pass time for each application, along with the amounts in the tank mix, and some basic information about the weather conditions and whether it rained within an hour or two after the application was made, or if the rain occurred much later or not at all. And then information about the sample collection would also go into the tables, the timing, and what exactly was collected, what kind of modifications were made. For example, I mentioned cutting the samples into quarters, possibly into peel and pulp. That would be described in these tables. The report should, in the summary, include an indication of field trial separation. That is a very important issue for anything submitted to JMPR. So it's that 30-day separation or 30-kilometer separation between field trials that are within a country, and any other variables that might be useful to help separate the trials. Next slide, please. The analytical data is normally summarized in an analytical summary report that is completed by the laboratory research investigator and signed by the laboratory research investigator. So that is an example of a contributing scientist report. And that would be included in its entirety in the final report. It has a brief description of the method and the results, and it has tables with the residue data and recovery results. So the recovery results, if you're not a chemist, that is when at the laboratory, they will fortify a control sample with a known amount of the reference substance, and then immediately do an analysis of what they have just fortified. And ideally, they get back 100% of what they have fortified. But in reality, they may get 80% or even 110%. It rarely comes back exactly at 100, but typically between 70 and 120% is considered an acceptable range for recovery results. So that information is included in the analytical summary report, along with the residue results. How many parts per million have they found in the samples that were treated in the field? And if freezer stored stability data is required, then that is reported also. That is where at the lab, they fortify samples. They fortify some control samples as soon as they've received them from the field and put that in the freezer. And then when they are finished with all of the treated samples from the field, then they would analyze the freezer samples to ensure that the chemical is stable in freezer storage during that time. That is only required when there is not already sufficient data available on multiple crop types for that particular chemical. And that please mute your microphone and I'll continue. The text of the final report will include some of the most important details from the analytical summary report, but it is not necessary to transcribe all of the information. Next slide, please. So after the report has been printed and the study director and the sponsor have signed it, and it includes the signed quality assurance statement from the auditor, then the printed report is archived. A scanned copy of this report may be submitted to JMPR if that pesticide use has been scheduled for review. There is also another report that is required in a particular format for JMPR submission that further summarizes what has already been summarized in the final report for each study. But the JMPR report would possibly include summaries of other studies with the same chemical, but on other crops that have been completed and which have been scheduled for review. Next slide, please. And that is how a final report is prepared, or what needs to be in the final report from start to finish. And I thank you for your attention.
Muchas gracias, Ken, por esta
Thank you,
presentación.
Ken, for your presentation. They say that time flies, And I think that's completely true. We're nearing the end of today's sessions and the final presentation of 2 days and 8 hours of technical exchanges and exchanges of experiences between countries. I was looking at the chat and I saw some questions about the presentations. We need you to become the #1 fans of the website, of the foundation's website, because all of the presentations, all of the videos, and all of the information that we gathered in these 2 days of workshop, you'll be able to find at the website of our foundation. So I will like to ask my colleagues to please write the address, the URL on the chat, so that all of those who have never been there can add them to their favorites. You can also search Minor Use Foundation and you will find it very easily. And you'll have at your disposal 8 hours of training from these workshops, including all of the PowerPoint presentations as well. So we're getting to our final presentation for today's session, and this one is about storage and preservation of files, in charge of Veronica Picado, who is a consultant for the foundation. So Veronica, I would like to give the floor to you, and thank you. Good morning, everyone. When I don't know if you can hear me.
Presentación en español. Okay. Vamos a llegar al último principio.
So we're getting to the last principle. There are 10 of them that we have looked at 9 of them already. On a very personal note, I was thinking about this yesterday and today. I was thinking about how cautious we are while speaking, because I was making sure that the battery of my computer was full, that my cell phone was there in case I lose my internet connection. And I was thinking that at this workshop, if something happens to me and I cannot speak, any of the presenters from yesterday's sessions or today's sessions could speak for me on my behalf. So I'm very happy for sharing with all of these members of the panel, and I'm happy because I know that all of them are prepared to talk about the principles, the GLP principles. It's great to be part of a group like this one in which anyone can speak. So as I mentioned, my name is Veronica Picado. I'm a consultant for the foundation, and alongside Adriana Castañeda, we promote the projects in Latin America with the technical support from Joe and Ken. A very important part that I hope you be able to take with you from this workshop is the objective of these GLPs, which is mainly reconstructing the study. I know that we have many people here who are joining us from labs and who are used to working with ISO rules, especially with the 17025. If this is your first encounter with GLPs, you may have some questions about the difference between this ISO regulations and a quality system for GLP, for good laboratory practices. To me, the main one is that with the GLPs, we aim to have or to keep a traceability and to be able to reconstruct what happened exactly when that study took place, assuring the quality and the integrity of the data that was collected at the time. With the 17025 ISO regulation, we aim to provide a technical relevance to the trial. We're not aiming towards trial relevance, or this will be determined by the regulator. Obviously, the studies need to comply that, but it's not part of what the quality assurance reviewer will try to verify. What the GLPs try to do is that if they want to come 5-10 years after and verify what happened, they'll be able to find the data, they'll be able to interpret the data regardless of the fact of regardless whether the personnel of the study are there still or not. So it's very important to maintain this in a way in which that it's perfectly preserved, because this would be a serious deviation if these are not kept properly. We wouldn't be able to have a successful study and keep all the data, and when sending the final report, not know what happened with the field notebook, what happened with the chromatographers at the labs, to know everything that has to do with the personnel of the study. If it's not there in the including in the GLPs, if it's not included, there's nothing for the regulator to check afterwards. So this is one of the main criteria that need to be kept in the long term, and we need to comply with it day by day until we can delete whatever files we have stored. This is one of the criteria that is that are the hardest to meet, especially because of the amount of data that is produced. The OECD has a reference document. The code of that document, you can see there on your screens. If you Google it, or any other search, or using any other search engine, you'll find that document. Many of you have been asking, where can you find the principles or the reference documents? So I would like to explain briefly. The OECD has a principle document in which it enlists the 10 principles, and for those that require a little bit more support or further guidelines, they have a separate document and that provides these guidelines. One of these principles is the storage, and where it explains how to meet this principle. It starts by defining what an archive is and what or archive, and the person who's responsible of the responsible administration and storage of that data that was generated. Because having a study, and if you cannot define who the main field investigator, the study director, or the lab investigator, yes, you can define who that people are, but you need to define who the archivist will be and who will keep all of that information in accordance with the GPL, the GLPs. This is not only after you finish the final report and when you store it, because data may be lost during execution. Trials or studies can last for 1, 2, or 3 months. And anything can happen during that first month to the documents. So it's important to apply this principle since the start of the study until the end of the study, not to lose the traceability of this data. So this is a responsibility that involves us all. It's not about hiring an archivist and that person will be in charge of storing the documents and it's their problem. No, everyone has a role, an important role in terms of ensuring ensuring that that information is kept. So starting with the sponsor, of course, they need to make sure that the data is preserved and that there's continuous access to them. If afterwards the monitoring authority of the regulator comes and asks for an inspection of the trial or the study, the data must be available and easy to access Yeah, because it cannot be the case that they tell JMPR, EPA, no, you cannot perform the inspection because we don't know where the documents are. This happened 5 years ago, so we don't know what happened to these documents. No, they need to have access to these documents, and we need to know where that is, and there's a person responsible for that. The facility director or the facility management needs to be responsible for the provision of archive facilities. The protocol of the study plan that Edwin talked about says in the last section where those where that archive facilities will be located, so that everybody knows, the sponsor, the study director, and all the personnel know where these documents and this information will be finally kept, and even samples, because we will look at how the samples need to be archived. The study director needs to ensure that they are complete and transferred to the archives. A principal investigator is responsible for or needs to make sure that he and their personnel are filling out all of these logs and the field notebook, of course, and once they finish their part, that this is transferred to the archive or whatever is indicated in the protocol. So this is a responsibility of each principal investigator. Quality assurance needs to inspect all aspects related to this principle. They will inspect abiding by the 10 GLP practices. So they put this aside. They need to inspect how this is being kept, if they're meeting everything that's said in the GLPs. This needs to be part of the inspections from quality assurance. And hopefully this can happen before sending the final report, and not many months after, when they realize that it's too late, and since they already sent the final report, the documents have disappeared. And the archivist is the person responsible for file management and then everything related to controlling that data. What do we need to have at those archive facilities? We'll have a brief presentation, but I really would recommend to read this reference document that I've mentioned, because this is a very complete document. This is one of the most important criteria. This is what will allow us to keep everything in the long term. First of all, well, it will prevent unauthorized access, because we cannot allow for someone to come there and who thinks that this can be deleted or thrown away, or that someone wants to change a page. Anything that can happen. Access needs to be controlled all the time. The archivist or the person in charge, the director of the facilities, need to designate and to decide who can consult the raw data, the archive procedures, the curriculum. I cannot come tomorrow to Senasa in Peru, say like, okay, I want to look at how this or that is being handled, because I wouldn't be authorized. So the person in charge should be able to tell me, no, you're not authorized. And people shouldn't be able to take this with them. They can look at it with alongside the person in charge. There needs to be a control kept of the time and date when these are consulted. This should be able to resist local weather. If it's a cold or a very hot area that can deteriorate this archive, we need to control. In general terms, these rooms have controlled conditions. They need to be able to resist pests, insects, rodents. So again, this needs to be a controlled environment in order to keep this for many, many years. How many years? OECD recommends whatever the regulator says. So depending on regulations in your country or the regulator that you'll be submitting these studies to, many say that for at least for the time that the commercial product will be available in the market. But again, you cannot delete or throw away anything without an authorization from the study director or a superior if the study director is no longer available. This need to be protected against fires, and when necessary, there must be backup electrical power. Which cases? For instance, in the studies that we carry out in terms of establishing the maximum residue limits for grains, we need to preserve a frozen sample, and we need to ensure that that sample is kept at the corresponding temperatures, which would be below 16 degrees C. So those freezers need to have backup electrical power. Next. So what are the conditions that we need to have, the archive conditions? I already mentioned controlled environment, the conditions of the environment. We need to have an SOP. I love this picture. I took it from an IR4 presentation. You can see that they have an alarm. This is next to the freezer where the samples are kept. They have a phone dialer. They have emergency notes. They told us during this presentation that they get alerts on their cell phone, and the procedure says that if you get an alert, whether on your cell phone, on your email, even during a weekend, if it says that the samples are at risk of defrosting, so we need to have that very well organized in our archive facility. I would like to, there can be a trial site in the field and one at the lab. And then we have the facilities, which is the whole place where the study is taking place. So we could have the archive and then a testing site. This doesn't mean that the field investigator doesn't have to have a place for storage. This could be a metal locker that has fire protection, or something that can prevent that someone can grab a page from the field notebook, for instance, by mistake. So yes, we can comply with this criteria. It doesn't have to be as sophisticated as what we're seeing right now, but yes, both the field investigator and the lab investigator need to ensure this. They need to comply with this criteria, perhaps with simple conditions, but they need to meet this archive conditions that are requested at the GLPs. These are some examples. These are photographs from IR4 as well, and how they archive. This is a picture that I wanted to show you because it's a great example of when you need to archive and you are working on several studies. What do the GLPs call for? You need to index them. So what does it mean? To keep an order of the data in a common way that allows me to search for them very easily, because in 10 or 15 years, an inspector may come wanting to check the data for that study, and perhaps we will no longer be there and we won't be able to tell them, yeah, this was kept in aisle number 3 or freezer number 4, perhaps there will be a different personnel because the other personnel retired, and that new personnel who know nothing about the study on papaya have to be able to tell them, yes, this was archived, and that archivist needs to be able to find this in a very easy way and to show all of the data, all of the relevant data. So again, we need to keep this in an orderly fashion with an index that will allow us to identify this in a quick way. Some examples that I have seen that they keep some of this file lockers that we've seen, we call them R2D2s in Costa Rica. So they keep a number for the folders and then for each one of these lockers. So they look for each document depending on the number of the locker and the number of the folder. Each one of the entities that we work for handle things in a different way. So we can do this in different ways as well. But the most important thing is that these are kept in an orderly or following an orderly procedure. So there, we need to have archives of this test substances, and the laboratory materials that were used for the analysis. All of these things we need to keep. It's not only about documents. In case that we have electronic logs or records, we need to ensure that they are safe. Yesterday, there was a question that was asked. Someone wanted to know if all of the procedures and the logs could be kept electronically and only electronically. This would be very difficult because you need to make sure that you recorded the data at the same time when you generated that piece of data, and that it will be preserved for a very long time. So yes, if you're keeping these records or logs using a tablet at a mango or banana crop, because in Central America, for instance, these are in very distant areas with no internet access, with no electricity. If you don't have how to charge your tablet, you may lose information if your battery dies on you. So it's important to keep a written log of this for the long term. Perhaps there are certain archives that are very long, for instance, the results of the chromatographers or Excel spreadsheets. The OECD permits to keep this electronic logs as long as you can ensure that this data can be consulted over time. I'll give you an example. When I went to university, we used to we used floppy disks, and if we had those floppy disks nowadays, we wouldn't be able to find a computer nowadays where we can check that information. The computers that we use today don't even have a CD port. So if you keep your electronic logs, you need to keep in your procedures how you will be updating them to more recent technologies. You need to keep a record of which information was transferred, and you need to ensure that year after year, this data will be available electronically. If you kept the raw data from the chromatographer, and if the headquarters of the chromatographer or the manufacturer no longer uses that software, that will be a problem. So it's better to print out that data if you use Excel spreadsheets. Also, it's important to print that out so that it can be visible. However, yes, the reference document allows you to keep this, but it's important to make sure that you'll be able to ensure reading, access, and the continuity.
In the case of wanting to eliminate these, then there must be a registry of what was eliminated, when it was eliminated, and basically a permission. As I mentioned, what the OECD document recommends is to do this according to the regulator criteria of when these can be eliminated. And the document does recommend that there might be at least 3 inspections of the data before those being eliminated. Next, please. Well, here briefly, the reference document has some guidelines or recommendations of what should be included in the operating procedure of that file. Things that should not be left out of how the access to the files will be, the definition, description, the index methodology that's being used, including the indexing of the electronic registries. All these registries must fulfill all the requirements for the file that happen on file. The ensured of climate conditions, controlled access, everything that's done on paper is done electronically. Under what conditions these must be kept, both documents and materials. With materials, I miss, for example, samples, standards, and other things of that type. Aside from electronic registry, the processing of data registry when, for example, the field notebook is moved, or when the report or data are moved, they go through a custody chain where the people in the lab formally deliver these, at least in my experience. We have used even saying, given so many pages, and sometimes it's very detailed work of what they are delivering, because the person, the archiver, is going to be responsible for them and not basically losing half the file or the report or the data in any part of the procedure. So if they don't have a procedure of this type and they start to create it with this document, they can have some guidelines as far as what parts need to be included, so nothing is left out of how to perform this process. Next, please. What should be archived? All the samples, the documents, raw data. What happens, for example, if the archive where we have stored data for a long time and everything relative to the studies is closing? I'm imagining something drastic. For example, they work for so-and-so institution, and for some reason this institution is going to disappear. So they're closing all their different departments and they don't know what to do with these documents because this is not going to be their occupation anymore. Or maybe a lab that is used to performing these type of services, and maybe they are selling the lab and not offering those services anymore. So what are they going to do? Well, they need to be transferred to the sponsor. And of course, we need to let the people involved know that that process is taking place, and that from this moment on forward, the documents will be in hands of said person or said organization responsible. But what's correct is to give them to the sponsor. For people working with us, the sponsor is the Minor Use Foundation. Next, please. Very well. So it's something to expect that we have program, list, raw data, interpretation, evaluations, all the documented parts of the process. If there are WhatsApp exchanges, now that's very popular among us, and we feel that to be even quicker. So even to share and to print out what we speak about with respect to the studies, and this should be stated as part of the process. The SOPs, the people involved in the study. We need to show that the staff was properly trained to be able to execute the functions that were assigned, and they were trained in good laboratory practices. Here are two types of documents that we can archive. First are proprietary to the study, and the other have to do with the facility. There will be data in the field that are belonging to the specific study, but there will be other data that will be falling in common for all the different studies that you're performing that have to do with the environmental conditions of the lab, or the samples, the temperature registrations from the freezer, or the use of the scales, if these were used. Maybe they were used both for banana and papaya studies. So there can be a classification that apply for more than one, or registries that apply only to one of them. The data from the staff involved, the CV of each one of them, the certificates may be stored in a common way with a correct indexing, so these do not need to be replicated back and forth for each one of the studies that go into archive. Next, please. Well, briefly, the principle works very well. I hope this has served you well, and thank you very much for your participation and your attention. And I leave you with Eric. Thank you, Veronica, for this very complete presentation. Before we go on to the Q&A section that will be managed by Veronica, as we did yesterday, I allow myself to inform you that in chat, I want to verify. I would ask Adriana to help me. I don't know if we did this already or we set this again. I don't know if we did this, we can do it again because there's a lot of speed in the way that the chat is going. We will put in chat a link, and this link is meant to fill out a electronic format, a survey basically that will help us evaluate this event. I have asked that you write down your names, your institutions, your countries, and this is helping us significantly. But this survey that we're sending out, I'm seeing it right now in the chat, this will help us not only to be able to evaluate the event itself and to have criteria to be better, but it will also have in a process manner your information with your corresponding email. The email really is very important for us to be able to maintain communication with all of you. Further on, we will have more events with new products of cooperation, training, information, news that the foundation would like to share with all of you, and we would like to ensure that we can be in contact with all of you if the need arises. I would ask Adriana to put in chat a couple times to leave the link present. So please copy this link, open it and leave it on your browser on standby if you like while we finish, and we will thank you very, very much. We will thank you very much that you please fill out the survey as an evaluation for us. We will be typing out this link permanently in chat while we go through the Q&A section to ensure that we have this information available. I would not like to keep speaking because we are very short on time, and I would just like to continue with Veronica and to begin with the Q&A section. Veronica, please go ahead. Thank you, Eric. Yes, briefly, with respect to the survey, if we can make use and write down the name and the email where you want the certificate for participation to be sent, and the name as you want it to appear on the certificate, because in chat some have maybe put their name without their last name. So let's go back to the question. I'll go back to questions from yesterday that I think it would be good that we all listen to the response. Those that we are not able to answer here, those will be answered by email, just in a more personal manner. Veronica, I'm seeing here in chat that Dr. Drust, the president of the foundation, Mr. Drust, is interested in trying to share some words with the group, but he has to leave. So we will vary the protocol somewhat, and we will, Mr. Drust, speak. He's the president of the Minor Foundation, for him to exchange a few words with the group, given that he has a tight schedule and he has to leave. Thank you, Derek, for being here. And we've had some very, very interesting sessions, very rich sessions, many connected countries. Please, the floor is yours.
Thank you very much, Veronica. I appreciate the opportunity to talk with all of you today. It's a pleasure on behalf of the Minor Use Foundation to have been able to have this workshop on the principles of good laboratory practice. I'm very, very happy to see the participation that you have today, and also to learn from the conference organizers that there were more than 500 people involved in the meeting yesterday. This is really exciting to have so many participants from at least 21 countries in Latin America, the Caribbean, and Africa. This has been an excellent opportunity for you to interact with other regions and countries and share input and ideas. I've been listening now for the last 45 minutes to the presentations, and these are excellent presentations that these presenters have prepared. We congratulate all of the presenters on these excellent presentations. And as you know, they're representatives of partner countries, and all of them have benefited from previous trainings like this, and it's allowed them to be train you and extend their knowledge to you. So this is a wonderful outcome, and it's a goal of our foundation. It's to train the next generation who can train the next generation to maintain scientific expertise and credibility and build it at country and at region level. We hope this workshop has met your expectations as well. And as has been mentioned by Eric and others, the presentations that have been given today will be shared on the Foundation's website. And I know that you saw the link to that earlier in the chat and on the screen. You'll find the presentations there. So I don't want to take up your time for questions and answers. I very much appreciate the opportunity to speak with you today, and I wish you well in your Q&A session, and I wish you happiness in your work. On behalf of growers and other stakeholders, thank you so much, and have a wonderful day.
Muchas gracias, Dirk. Entonces,
Thank you very much, Dirk. As I mentioned, we will go on to the Q&A, and we'll go back to some left from yesterday. I think it was from Peru, where they were asking if we may separate the test sample that was used on the field in a separate jar from the original to move this or to be measured or weighed directly in the lab. I don't know if some of the panelists or the presenters want to make reference to this question. I think he refers to times when there is hesitation or caution of not taking the whole sample to the field if anything would happen on the transportation process, but just taking the necessary amount, or maybe a couple of replicas, but not as a whole, having the risk of it suffering harm in the process. I don't know if Joe, Javier, Ken, maybe Edwin, somebody would maybe like to take this question. Pero, can you hear me? Yes. Well, this is a topic that's quite hard because the way that it should work is that the substance arrives as a whole at each one of the trial locations. But nonetheless, we have had situations in Colombia because we need to move to areas by plane where these trials will take place. And on planes, they don't allow us to take a liter of the substance with us, and even less if it's in your handbag. So one of the options that we have implemented is to request the owner of the sample that when they send the sample, they send that dosified already in jars, where we know that with one jar, we can perform one trial. So that's the case of espinetoral in pineapple. We asked the company that if they can send it in that way already in the correct dosage in a jar, and that's the way it arrived. It's very important that all the jars must have all the information of the test sample. It needs to have the lot, the expiration date, the name, the ID number, everything that refers to that sample. I would like to maybe ask Joe, because he may be able to clear up what happens when, for example, we have a liter of solution and it's already been received by the field researcher. So if we can dosify that ourselves or not. Joe?
Yeah, thank you, Edwin. What you said is absolutely correct. The registrant often does not send small doses. They will send you a liter. So if they did, that would be fine. It is permissible to transfer out of the main container the amount that you need for your application and take it to the field. And as you said, as long as it is properly labeled and that you do have a temperature recording device with the test substance, so you know that it has maintained integrity during transit. There are two things I want to mention in addition to what you just said, Edwin, and that is it may be wise to take a little more than you actually need for the application in case there is some spillage when you get to the site. So you want to make sure you have enough to do the application, especially like in your case with the cacao, you have to travel quite a distance to go to the site. You don't want to spend hours and a plane ride getting to the site with the exact amount of test substance and then have a slight spillage and realize you don't have enough. So it is permissible to take a small amount if it's properly labeled and take a little bit extra or maybe even a second dose in case something happens to the first one. On top of that, I want to caution anyone doing this that if there is any test substance left over once you take it to the field, do not put it back into the original container. It will need to be disposed of out in the field or some other method safely disposed of. There is a chance that it may have been contaminated in the transferring process, so do not add back to the original container. I hope that clarified and illuminated a little more what Edwin had to say. Ken may have more opinion about that, but
No, I agree with everything that Joe and Edwin have said.
okay, thank you, Ken. And thank
Gracias. Vamos a pasar a otra pregunta.
Thank you. We will move on to the next question. And here I invite Carla Arrieta to answer it because it is directed specifically to the SOPs that she explained. In the case of the phytosanitary service from the state following the guidelines of ISO and the state, the documentation system for these is complementary between them, or is it managed in parallel to be able to fulfill the requirements of both guidelines? Where our case, we do that in parallel. When there are certain principles of the best practices that are shared with ISO. So what we do is that since these studies really take place 1 or twice a year, it's not that's continuous as in the case of ISO, then certain information is shared or is complemented with one from the other. So we don't need to duplicate registrations and documents that are already inside the ISO certification. So one complements the other, or at least that's our case. I don't know if that answers the question. Thank you, Carla. I take the opportunity because we just finished the matter of archives. Archives require that we save the original files or a truthful copy of these original files. I know Carla's experience well, and one example of managing these parallel systems was the certifications of the lab standards of the reference material or analytical standards, or the calibration certificates of all these different sets of equipment. So we said, well, where do we archive these? Well, these, according to the ISO and according to the legislation in Costa Rica, these are stored for 5 years, or are they stored according to the BPL? And Carla decided, well, and he said to the people generating these, the originals that were originally archived. And this is an advantage of having these copies saved. I don't know, but basically a truthful copy of anything that's being archived. There's another question, and I think it might be the last one. I will revise, maybe there are some that are important that everyone is clear on, because it might be questions that people have in common. Maybe we could share many that are important to everyone, and the responses when we send those out will be sent out with the participation certification. It says, we have identified, this is a question from Mexico, we have identified situations on the field or during the development of the study that imply a level of deviation that leads to a suspension of the study, or are they just written down and continue with the study? So I invite Ken or Joe, maybe Saul, to explain how we manage these deviations when they are presented.
Yes, I will respond. Whenever a deviation to the protocol occurs, it must be communicated to the study director as soon as it is possible. And even if you don't know that the deviation has happened until several days later, perhaps once you know it, you should contact the study director. And then it is up to the study director to determine if the trial may proceed exactly as had been planned, or if perhaps some change must be made to what is required going forward in the trial. Or in the worst case, it may be that a deviation means that the data from the trial will not be useful and that the trial should be terminated. But always contact the study director as soon as you are able to.
Gracias, Ken. Creo que Saúl quiere agregar alguna cosa más.
Thank you, Ken. I think Saul wants to add something else. Yes, thank you very much. Usually, when we try to select these plots, we allow these to have certain communication as far as it's possible, because the deviations due to climate change might be very common. And it happened to us because we began working in the cold time of the year in our application. Our protocol had a maximum allowed temperature for application of 31 degrees C. And in the last application, before we began the last application, the field researcher was just about to go through the process, and it went from 30 to 32 degrees. So we immediately stopped. And since we were in the last application, which is basically the most important, and being fearful that the temperature would not go any lower, we got in touch with the director through a WhatsApp call. And we tried to communicate this because it was possible for him to be knowledgeable of this. And he said, well, I know what happened. Let me talk to the owner of the molecule or the people necessary to consult if this could be a subject to an invalidation of the trial. So the decision that was taken at the time was to wait. Some things cannot be done and then be registered as a deviation to the protocol, because this could be a serious deviation that put a compromise. And they said that it was necessary to wait to have the necessary temperatures for the application. And this might happen. And that's why it's always necessary to be in constant communication with the study director, both before beginning the application and to be paying attention of any conditions that might change. There are other deviations, such as the one that I explained with the weight of pineapples, which was a deviation, but it did not compromise the integrity of the trial or the data validity, because these are factors that cannot be controlled. But immediately, I was about to commit a mistake because I was supervising the people in the lab while the sample decreased, and the protocol only allows me to get to a certain point. And since I noticed that the sample was too heavy, then I will continue to cut in 16 parts. But I consulted the director first, and he said, no, the protocol only indicates cutting to 8 pieces. If the piece is too big, then there's a deviation that must be stated. So the message is basically to have constant communication with the study director supervising the process. Thank you. Thank you, Saul. And very well, unfortunately, this was the last question. Given the time constraints, as I said, we will answer all your questions through email. Before we leave you with Eric to finish for today, since we don't have a registry, the only way that we have of sending you a participation certificate is that you write your information down on chat, your name and your email, or that you fill out the survey. So the people that have not done this and that would like to hear when these information is all available online, even the responses to the questions, then we invite you to write down in the chat box your email and your name, and maybe the organization. Thank you very much. Thank you, Veronica. I see in my phone we have 10:01 Costa Rica time. So therefore, we are doing very well with time. We have fulfilled the time frame that we had scheduled for each one of the presentations. And globally, I have nothing else to say but to thank you all for your participation. It has been very important to listen to the words of Dirk Trost, the president of the Minor Use Foundation, and also a closure of the event. I am sure that this is a process that is just beginning. We have a whole team working on a training curriculum that will reach ambitiously and aggressively all the different countries where we are intervening. For our organization, it has been a pleasure to collaborate with the foundation and to be in contact with all of you. It's a pleasure. Please remember, anytime that you require any type of help from our side, you can knock on our door in the different offices in each one of your countries. I would like to thank in particular each one of the presenters, the panelists, the countries, to be able to exchange our experiences and knowledge that has been developed throughout the period of time. This has been very enriching, and really it has been very hopeful because we are receiving training from people that not too long ago were trained themselves. So I think it was Dirk that said this. We are in a process of continuous training where new generations will be incorporated to this line of knowledge. And really the main objective is to have many more trials in our countries. Given the productive characteristics of our countries, we need to have more and better phytosanitary solutions for our minor crops. And the effort that's done through the foundation and through all of you is very important. I would like to end again by highlighting what Veronica mentioned. It is very important that you fill out the survey. This will be very important information for us. I would ask Adriana that you please put the link in the chat again, and the platforms English, French, and Spanish will be open, so you can copy this link and help us by filling out the survey. I have nothing else to say but to say good night to the people in the Africa region. I think they're already getting to that time. From my side, I am about to have breakfast, but thank you very much for dedicating 8 hours of your precious time towards participating in this event. Thank you very much and good day.